No, still 3 minutes to go.
So how does this work? They're now in this breakout rooms where they discuss the previous.
Well, the breakout room were the speakers essentially.
But weren't you the speakers today? You were talking about film phasers today.
Yeah, so I have 4 or 5 people in the breakout room, but yesterday I have 9, 10. So it's okay, you know, it's simply, you know, most of the questions are technical. So
well,
I see
that's
that
good.
that example matters very little for everybody. Everybody's interested in the method and how you do the method and so.
Detail oriented. That's good.
Well, it is okay because this is the purpose of the. So, but most of the questions were about method.
Huh?
Okay.
Well, they're going to hear more about method now.
Yeah. So you will talk about method, and actually people ask if you can use. You know, one of the questions I have, if you can use the sine and cosine filter on a wide field microscope. Of course, doesn't matter, you know.
Well, can you hear me?
We can, yeah.
Okay, perfect. So we are almost 2 minutes to go. So I will start, maybe Alex, if you want, you can already share your screen so we can check that everything is going okay.
So you are activated.
I'm here. You can see this, right?
Yeah. Yes. So let me introduce you. Well, so I have now the pleasure to introduce Dr. Alexander Barmishana, who is a postdoc at the Laboratory for Fluorescent Dynamics and the Biomedical Engineering Department, the University of California, Irvine, working with Enrico. Alex is a world expert in imaging processing, programming, as well as artificial intelligence techniques for analyzing imaging. He has a long experience on this kind of technique, and when he joined the LFD, he used most of this background to basically share the experience and met the experience with the spectroscopy on images. And I think his work is really pushing the frontiers of the film and phaser analysis. And today he's going to talk about this multiple component analysis. As I just commented before, the possibility to analyze 3 and 4 components, sometimes without even knowledge of where are those components. And he's also pushing all the stuff related to artificial intelligence combination with film to analyze the data. I think this is really a huge area to grow, and he will make really big contribution. We spend a lot of time together since we basically share our office at my time at the LFD, and I always enjoy our fruitful discussion. It was always a good person to discuss about imaging analysis and all this mathematics, and we enjoy also playing tennis during those days with Bautista. So thank you very much, Alex, for the talk today, and I'm very happy that you can share with us your presentation.
Thank you for the introduction. And yeah, the conversations weren't only on image processing. I also enjoyed all the biology you had to, and the biophysics you had to teach me. So yeah, it was a good combo that time. We keep
a compliment.
Keep getting this. I guess I can admit everyone that is just appearing here, right? This is just.
Yeah, I am.
Okay, you also have control of that, right?
Okay, perfect.
So yeah, you've already had 3 sessions today about the phaser approach. So, but I'm going to do a introduction nevertheless, and I think that it's good that you have different people talking about the same subject, because you will basically the ideas will get into you more easily. This is going to be a pretty math-heavy discussion. I hope that people don't get scared about this. And Enrico was saying that in some of the breakout rooms, people were asking about the details. So I think that maybe you can ask about the details here if you have any questions. So yeah, please interrupt. If I don't see the chat window down here because I'm on full screen, but if someone asks a question on the chat, Leonel or Enrico, whoever, please interrupt me and I will gladly address it. Okay, so the phaser transform in fluorescence microscopy. You all know that you excite your molecules to some excited state, and the emission is what you collect as fluorescence. and it's usually of lower energy because the gap is lower energy gap. So there is the typical knowledge you have is that you excite with, say, blue and collect in green, or excite in, so you have a redshift in the emission. So there is a spectral quality of these photons, but there's also a lifetime process. So there's a time typical time of the fluorescence that is occurring in this molecule. This depends on the shape and the orbitals of the chemical compounds that are emitting the fluorescence. So it is a characteristic that is unique to that particular molecule. So in fluorescence microscopy, you can collect the color of the photons, and you can also time what time they arrive. Now, to do this, you need to synchronize your device to some pulse source or some modulated source. It's easy to understand in a pulse source. So you have some laser that is exciting your samples at a certain frequency, and you measure the arrival time of each photon after each excitation. Pretty straightforward. Now you've seen the phasor transform. There is this analogy behind the spectral and the lifetime phasor transform, because in both cases, what you have is a photon histogram distribution, in one case as a function of wavelength, and in the other as a function of time. So how many photons I am collecting in each color. In this case, we have little in the blue, many in the green, and not so many in the red. Or how many photons I'm collecting at each arrival time. So in this case, we're getting all the photons nearly at very short times, and then you get less and less and less and less. The phasor transform basically maps this shape of the photons that arrive at each pixel to one position on the phasor plot. And as you can see, the equations are very similar. And what you get is two coordinates, two numbers to map on the 2D plot. And in the lifetime case, it's basically the same. And then the position on the phasor plot can be then used to, for one thing, you can color the original intensity images using depending on the position of the phasor plot, but you can also extract meaningful information depending on the position and how the stuff clusters on the phasor plot and so on. So details of the phasor plot. As I said, you're exciting your sample with a pulse laser source, and you're collecting the individual time each photon arrives. And by accumulating many, many photons, you build your photon histogram of arrival time. So this corresponds to the stimulation period. Typically, our lasers are operating at 80 megahertz. So you are stimulating 80 million times per second, meaning that the time between pulses is 12.5 nanoseconds. So that would be the stimulation period is 12.5 nanoseconds. So you would stimulate at one point and you collect all your photons. Note that this is, now we're talking about 10 to the 5, 10 to the 6 photons. In reality, you typically are measuring stuff at, say, 1,000 counts per second, 10,000 counts per second, 100,000 counts per second. That's what you usually collect in your microscope. So this is to say that basically you are exciting 80 million times per second, but if your collection is only 80,000 times per second, you basically receive one photon for every 1,000 pulses. So your photon density is very scarce. It's just that you collect for enough time to have a meaningful statistics. But in each pixel, your photon histogram would look more like this, like you have 1 or 0, or in the bulk you'd have 2, 3, maybe 4 counts. And this is what you're usually dealing at each pixel, not so much this. So at each pixel you have your distribution, you map it to the phasor plot, and it may look a bit scary, this, but it's only two functions here. You have your intensity as a function of time and a trigonometric function that you fit in your stimulation period. So it's the product of these two integrated over time. You will sometimes see an infinity here. You will sometimes see a period. Depends on, it's basically the same idea. I like to put the infinity because you're acquiring for long, long times. So your period is 12.5 nanoseconds, as I said, and maybe you're acquiring for 5 minutes or a couple of minutes. So it's basically you're accumulating the intensity across all this time. But you can also imagine as you grab all the photons, put them in a single period, and you obtain all the photon distribution, and then you integrate over a single period if you've put all the photons in the first period. It's the same idea. So you multiply by the sine or cosine function to get one or the other component, and you normalize by the total intensity. This is analogous to a first order term of the Fourier transform, but I like to see it as a trigonometric transform, as a mathematical transform by itself, the phasor transform. And here's a nice little animation to understand what's going on. So you have your intensity as a function of time. You fit a sine function in this case in the same period as I told you, it's 12.5 nanoseconds because the laser used for this case was 80 megahertz. You multiply the two, and in this case, well, you see that this part dominates and this part is kind of slower because it's a short lifetime, so to say, or everything is happening in this side here. You integrate it, so you sum the area below the curve. And this is a number that after dividing over the area of the black curve behind, gives you your S coordinate in the phasor plot. You repeat the process for the cosine, which is shifted 90 degrees, as you know. Multiply the two, obtain the integral, you obtain another coordinate. You now have your phasor position of this particular curve. It lies on the universal circle, but we'll see about that later. So now, what is this little n thing? Because 2p freq f, it makes the function fit one time. But if you have an integer number here, what you do is make this trigonometric function fit an n number of times. It's what we call the harmonic number. So you can basically fit the trigonometric function once, but you can fit it twice, 3, 4 times. So this is the same animation for higher harmonic numbers. Typically, the phasor transform, you only use the first harmonic. But since in this work we made use of higher harmonics, it's important to notice that you can do this basically. One detail is that since you're adding more and more bulks in your trigonometric function, the positive and the negative side tend to compensate each other. So as you go into higher harmonics, note that the phasor coordinates always shift towards the 0, 0 coordinates. It everything compacts itself down at the origin. Okay, now key concepts as a summary of the phasor transform. There exists the universal circle that has some expression, and it's basically a circle of radius 1/2 centered at the coordinates 1/2, 0. And this comes from the fact that if you impose that the intensity as a function of time is a mono-exponential, as we say, so this is a process that is, you have your excited state and you have a bunch of electrons in the excited state, and they have a probability of decaying to the ground state. The typical way to model this is an exponential. And if you have a single population doing this process, it's the same as a radioactive decay. You have your initial number of this A1, the initial number, and the exponential decay tells you how many you will have as a function of time. And at the end, you have 0. So if you plug this into the phasor transform, what you obtain is basically the coordinates of the universal circle. So it tells you that anything that is modeled like this will always fall on the universal circle. Now, here go the short lifetimes, and here go the long lifetimes, where long lifetime is this tau in the denominator of the exponent. So short taus, long taus. Note also that this is not linear with tau. This is like moves super fast from 0 to say 0.5 unity, 2 nanoseconds, 3, 5, 10, 30, 100, infinity. So it's the kind of the sweet area is this one here. That's why we use 80 megahertz, because many of the fluorescent processes occur in the nanosecond scale. So 80 megahertz is a sweet area to have from 0.5 to say 5 nanoseconds in the wide range. But everything else above 5 is all compacted here, and anything below 0.5 is compacted here. Again, now, so if you have in some pixel a process that is a combination of exponential decays, so some sum of n exponential decays, your phasor transform will map it to a position inside the phasor, the universal circle. I'm sure you've already seen this morning, but it's good to memorize, to remind it. The other final thing is that phasor addition follows linear algebra. So the combination of two components is expressed as a linear combination of the coordinates of the components to obtain the coordinates of the resulting phasor position. So in this case, you have one component here, one pure exponential component, another one here. And if you say that in your pixel half of the photons come from this guy here and half of the photons come from this guy here, you say 1/2 of the coordinate g of one plus 1/2 of the coordinate g of the other gives you the coordinate g of your resulting phasor transform at that pixel. So you're saying it's basically vector algebra. You have this vector that goes from 0 to the position, half of it, the same one, half of it, and the sum of these two vectors map to that position. If instead of half and half of the photons, what you're getting is say 1/4 and 3/4, you map the position to a position that is 3/4 away from the one that is less weighted and 1/4 away of the one that is more weighted. So these numbers here are what we call the fractions, which is basically the normalized fraction of photons that you receive from each of the components. Obviously, you have a range of any position in between. Okay, up to here. Any questions, any comments? I guess not. Yeah, important detail was that the fractions have to add up to unity. Obviously, you are normalizing the number of photons you get from your measurement. So the total number of normalized total number of photons is unity. You will see that sometimes I use omega, sometimes I talk about f, fraction, weight. It's basically the same. It's what the coefficient in front of the g and s coordinates. All right, so in the typical problem, you have some measurement on the phasor plot, and you know that your fluorescent species are of particular lifetime, so you know the coordinates of your fluorescent species on the phasor plot, on the universal circle, meaning these two here, and this is what you measure. The question is, if I know that in my sample I put these two species and I have my measurement here, what are the relative weights of the two individual components? And this is what we call the two-component problem, which, as you can tell, you have three equations. You only have two unknowns, which are the relative weights. So this is an overdetermined system. There's a solution, and you can even just do it by measuring the distances. You can basically do it geometrically, measuring distances on the phasor plot. So this is an easy example. No problem about this. The round example, exactly three equations, three unknowns, corresponds to the three-component problem. You have a triangle, and your measured data is somewhere inside the triangle, and the relative fractions of each of the three components can be solved from this system. You basically say express omega-1 as a function of the other, stick it in here, then isolate omega-2, and this is a system you can basically solve. Now, what happens when you have higher components? Well, then you're in an indeterminate system situation, and you don't have enough information, basically. You cannot solve, there's infinite combinations of the omegas that satisfy these equations here. And now here's when we go into the higher harmonic that I was mentioning before. So since the distribution of lifetimes across the universal circle is not linear, when you have the higher harmonics of each individual components, the polygon that results is not the same polygon, it's a different polygon. Which means that if you can measure your sample at higher harmonics, you basically obtain a new polygon and a new set of equations. The first intuition is that these equations should be the same, but as I said, it's not the same, because although the polygon seems similar, it is different by the way it's designed. So these equations really are adding new information to the system. So they do allow you to solve the system now. You have 5 equations and 4 unknowns. So you would have to basically obtain your decay curve, do the phasor transform for 2 harmonics in this case, and then you know the positions of the pure components on the phasor, on the universal circle. And because you know that they're exponential decays, you can theoretically calculate the higher harmonics of these. So the green points on the universal circle, you can calculate theoretically, which means that all the G's and S's are on the capital G's and S's are your data points, and the lowercase G's and S's are theoretical, that you know because you know what compound you're using in your system. So again, now you can solve this in general. In fact, you can solve it for any number of components if you add enough harmonics. Well, that's theoretically. So you will always have this overdetermined system, which in matrix form is basically a matrix containing all the G's and S's multiplied by your incognita, which is all the omegas in a vector, giving a vector which is the capital G's and S's, and the last one is unity. And you solve this by least squares, and you can find the solution that approximates the best. Yeah, key point. The harmonics of the pure components can be, that's what I was saying. You have a theoretical expression of the coordinates of the pure components and their higher harmonics. You can always relate the higher harmonics to the base harmonic, meaning using this relationship. Okay, so this was just maybe the end of last year when we were playing around with this stuff, and one of the lab members prepared these solutions and to test the system. So he grabbed 4 fluorescent compounds and in solution and measured the lifetime. I think I'll tell you later which ones they are. So he basically gave me this set of 7 compounds. He told me 1, 2, and 3 are the pure components. Tell me what are the fractions. So he didn't tell me these numbers. So as you see, compound number 1, it's basically full weight number 1, 0 the others. Compound number 2 are the same. 0014 for 3 and 0010 for 0010 and 0001 for 4. Now, the fractions correspond to, say, number 6 is exactly 1/4 of each. So it falls exactly in the middle of the polygon. It's this guy here. As you can see, number 5 has less of the first component and more of the other 2. So it's kind of shifted towards the right. And then there's number 7, which only contains number 2 and 4. It falls exactly on the line between 2 and 4. And does not contain the other 3. The question was, can we feed this into the algorithm and obtain these numbers, which a priori I didn't know? So I mapped all the positions on the phasor plot, individual measurements, and then this is the harmonic number 2, and as you can see, everything is rotated towards 0. And yeah, we ran the algorithm, and these are the results of the algorithm. It's distributions of fractions because you have many points in this distribution here. So it's basically images, 256 by 256 images of a solution, and you have some error there. And that's why you have some spreading on the phasor plot. It's not all falling on the same exact point. And the spreading on the phasor plot is what gives you a spreading in the recovered fraction. So as you can see, I feed it solution number 1, solution as in chemical solution. And you obtain fraction number 1 of unity, and all the others are 0. The same for 2, 3, and 4. And then, as you can see, number 6 was the one that was exactly 1/4 of each. You get 25% of each. Number 7 is the one that only had 2 of them. And as you can see, the results are neatly recovering the fractions. So basically, this was proof that it works. And we published this at the beginning of this year. And yeah, I think that's basically it. Yep. So yeah, after doing this work, I remember one day I went to Enrico's office and said, so just by adding more and more harmonics, we can solve more and more equations. So in principle, we can solve n equations, so n number of components. And he said, definitely not, because the spreading of the phasor statistics gets messier and messier and gets to a point as everything compacts to here, to the 0, you will not be able to resolve stuff. So anything higher than these harmonics, basically, there's no way you can extract meaningful information out of this. But then we thought that, well, maybe for a low number of components, we can not only find the fractions, but also work in a blind experiment. So we don't know the lifetimes and we don't know the fractions. Can we find the actual positions of the pure components on the phasor plot by adding more harmonics? And as you will see, it seems like for a small number of components, we can do this. So into the last part of the presentation, finding not only the fractions, but also the fraction and the lifetime of unknown components, what we call the blind component problem. So for 2 components, it's kind of easy to understand because it's very geometrical. We even showed this in the paper. So you have your data point and some higher harmonic. In this case, I think it's a third because some limitations we had. Second would be around here. So you have your data, 2 harmonics in this case. And you're wondering which are the pure components on the universal circle that generate our data. You don't know neither the fractions nor the lifetimes. So maybe they are here, these two, so they're about 50-50. Maybe they are here, and the component on this side has more weight. You don't know. So the algorithm is basically scan for possible candidates. In this case, I choose this one here. So this is candidate for lifetime 1. And now, if this is a component that is generating our data, by construction, the other component has to be exactly on the other side. So I simply trace a line across my data. And so this is candidate 1, this has to be candidate 2. As I told you before, we can calculate the harmonics of our candidates because they're in the universal circle. So we automatically calculate each harmonic and then trace the line across the harmonics. As you can see, the line across the harmonics does not go through the data harmonic, which means that this is not a correct solution of our problem. You measure this distance. So now if you shift and test for a new candidate, the distance changes, as you can see. So the algorithm is basically loop for all the positions on the universal circle until the distance becomes 0, or minimize the distance. Now, it's easier to say in words, loop across all the positions, but there are infinite positions on the universal circle. So you have to discretize the space somehow. You have to decide how many steps, how big these steps are. And this is critical, as you will see. So if the step is too big, you may miss the correct point. So there is a sweet spot in between how many points you want to put in so that the algorithm isn't too slow, because obviously if you divide this into 10,000 points, it's going to be much slower than if you divide it in 10. So in the general case, this was for 2, but for n-blind components, it will be basically the same. You have some number of harmonics on data harmonics on your phasor plot. You test for candidates and their high harmonics. You solve the set of equations as however many you have as harmonics. In this case, with 3, it will be 3 couples of G and S equations plus the last one, which is the sum of the fractions equals 1. So in this case, it would be exactly 7 equations for the case of 3. You compute the recovered, what would be the theoretical positions for these components here, and then measure the distances to your data. In the general case, you don't have one distance. You have as many distances as components. So sorry, as harmonics. So what we do is use this metric, which is the squared sum of the distance, so that if all the distances are 0, this is basically 0. But if the sum of them is higher, it's higher than everything. Anything that is not the solution will give a capital D, which is of a positive number. So now the problem basically becomes minimizing this D. So the set of candidates with the minimal distance D is the solution. So these are simulations we did at the beginning to explore the different cases. We programmatically generate points in the universal circle, random points, random weights, sum them to see what would be the position of the data, and then just feeding this point here on the algorithm, try to recover the 2 weights and the 2 positions. And what we're doing basically is dividing the distance on the phasor space into an increasing number of steps. So what happens if you divide only into 10 steps and only do a scan for 10 positions? Well, basically what you get is a very coarse value of D. The value of D is the distance you recover. So as you can see, clearly there's a solution here. There's a minimum of the D, but it's very coarse. And what you obtain is not a very good approximation. As you increase the number of points by which you divide the universal circle, you gradually find a more precise position of the final components. And as you can see, at some point, I think in this case is about 100 steps, it finally does, keep arriving now, it finally does converge. So as you see, in this case, we find this well of solutions down here, and our solution corresponds to, let's say, a low G for component number 2, which is this guy here, this G here, and a relatively high G for the other component down here. Okay, what happens if we go into 3D space? Well, in the 3D space, you have now 3 sets of Gs. So you can't really represent this as easily. You have to kind of do sections in the 3D space. So I'm plotting here the position of component 1 versus 2, 1 versus 3, and 2 versus 3. So we add more and more points to solve the system. We converge to the actual solution. And at some point, we find a solution. And you see there's a well here. Clearly, this is our solution to the problem. Now, as it's a 3D problem, the computational time grows as the third power of the number of steps. So this is kind of easily going higher and higher. And in terms of computational steps, our loop has to go cover all the 100 or 200 steps, I think it was here. I think it's even more on the 3 dimensions. So it's kind of getting slower and slower. Here's another example of the 3D space. A hard example, meaning that it's 2 of them are very close. And as you can see, it solves it correctly as well. Now going to the 4D space, now things start to get critical. In this case, I was giving it up to 500 points, I think it was, yeah. And it never converged. I got fed up. I mean, I wasn't prepared to wait for 2 days to get the solution, so it did not converge. And it does not converge because, as you see, you have this you no longer have a single point of a minimal. You have what we call the valley of solutions, which is kind of a very spread out region where there's a minimal distance. So this means that, for example, here, if a very small change in this G here will give you a very high change in this G here. This gives you a high error. So clearly, you cannot simply divide this into more and more because, as I said, then you spend a whole week to calculate a problem. Consider that you're doing this for each pixel in an image, and an image has, say, 256 by 256 or 500 by 500. So you have to solve this problem for each pixel in the image, and it becomes impossible at some step. So we decided this is a good idea just to understand what's going on, but it's really not the best approach in terms of solving the problem. You have to find a better approach to solve this minimization. So yeah, and this is an example of your curve with a valley of solutions, which in this case is only for 2 components giving you distance d. But in general, down here, you have 3n dimensions. So it's the g coordinates of one component, the s coordinates of the other component, and the weights of each component. So say if you have a 3-component problem, you have 9 dimensions on the bottom, and you have this surface on top, which is the surface you have to minimize for 9 dimensions. So the brute force approach is scan this 9-dimensional space, 12-dimensional space, whatever, and find the point at which there's a minimal in this surface. Intuitive, but not optimal in terms of time. Obviously, it makes more sense to simply walk down the hill. So you're sitting on the side of a hill and you look around and say, okay, down is that way. So I simply walk down the hill. And this is crucial because in this case, we simply walked a line. It's a straight path. So in this case, the total time of computation grows as some number powered to the number of dimensions. In this case, the number of dimensions, you don't care about the number of dimensions because you're basically going down a line. So it's one-dimensional problem. So this is critical because it means that in this case, you can generalize to higher components. So basically the 4-component problem just takes a little longer, not as in orders of magnitude longer. I don't need to tell you that the growth of the exponential is not comparable to polynomial time. So I mean, so yeah, maybe for 2 components, both are equivalent, or even the brute force sometimes is even faster than the gradient descent because you have to calculate the gradient at each point and so on. But as you go into 3 and 4 components, there's no way you can beat the minimization techniques as opposed to the brute force. So the general case to solve this for any number of components is obtain your function to be minimalized as a function of however many dimensions. So 6 for the 2-component, 9 for the 3-component, 12 for the 4-component, and so on. Find the derivative in each direction, so some vector of partial derivatives, and solve the system. Here's an example for 3 components. You have 3 pairs of equations, so G as a function of the G's, first harmonics GNS, second harmonic GNS, third harmonic GNS, and the sum of weights equals unity. 9 unknowns, which are the 3 Gs, the 3 Ss, and the 3 omegas. You know that the universal circle contains the position of the pure harmonics. So you have a relation of the pure components. You have a relationship between the S's and G's, the small S's and G's. You know that you can relate the higher harmonics of the higher, so the S's H2 and S's and G's H2 and S's and G's H3. So you can relate them all to the first harmonic. And you can fit in the last equation to put on one of the weights as a function of the other two. This is other two, yeah. So the final system for the case of 3 components has 6 equations and 5 unknowns, which are basically these equations here, which you want to minimize. So you do say, which are the weights that satisfy these equations here? So you basically grab the guy on the right and send it to the other left. So say this element, this term here minus this term has to be equal to 0. And at the same time, this term minus this term has to be equal to 0. And this has to comply for all the equations. So that's why we have the term on the left minus the term of the right squared, so that the negative values become positive. So the minimum of this D is always our solution. When this equals this, and this equals this, and this equals this, and so on, it's our solution. This is the function we've got to minimize. The gradient of this function is a 5-component vector, and it's a very long expression. As soon as you go into higher components, you have very, very long expressions. But this you can do, even you can do computationally. So there are methods to do this, to just look at the surrounding of your surface and find the direction where the slope is going down. So this is kind of relatively easy to compute. If you want to do manually, it is hard. You will fill pages of equations if you do the derivatives of each of these terms. Okay, so now going into real data. These are the same solutions I showed you in the previous case, in which we had 4 compounds with the pure positions on the universal circle. And in this case, Sasha, our lab mate, he prepared a bunch of different combinations, and he didn't even tell me what they were. So, well, you can clearly see that this here is most probably a mix of these two here, 50-50. But what about these guys here? You don't know if it's only 3 of them contributing. Maybe this one is not there. Maybe it is there, but it's in a low amount. You really can't tell. Can we grab each individual one of these and obtain which are the compounds on the universal circle that generate this data? So, well, the first thing I did is, since I noticed that 4 of them were on the universal circle, I used the non-blind algorithm, forcing the positions on the phasor plot, on the universal circle, sorry, and trying to find the fractions of the other ones. So to have an idea of what the solution was, what I was expecting to find, as in the fractions. But the important stuff is, can I find the lifetimes on the universal circle of each of them? And this is the data we used for this recent publication, like a month ago, I think it came out, in which basically we obtained, so the gray bars are the lifetimes of the compounds for couples in this case. So we have the 4 compounds, the mixture of 1 and 4, the mixture of 1 and 3, 1 and 2, 2 and 4, and so on. You have all the combinations. When you have trios, well, you only have 3 combinations. And when there's only 1 combination of the 4 together. And as you can see, we are recovering, the gray is the theoretical, the known, 12, 3.9, 1.1, 0.2. And the red bar is our recovered lifetime for each of the components, which, as you can see, it recovers it pretty well for 2, easy. For 3, you kind of get more error for the short lifetimes. And for 4, it starts to also diverge a little. But we found that there was a little trick that can be useful. That is, if maybe you know for sure that the lifetime of one of them, or maybe you have some idea, or maybe the distribution of one of them is very small. So you say, okay, if the distribution is very small, I can more or less be sure that the value is around 10, so I can fix it. In this case, we fixed it at 11.8 because we knew that was correct. And then this improves greatly the recovery of the other 3. So a bit of the conclusion is that theoretically you can, but as soon as you have higher number of components, you start to get very broad distributions. We can clearly see that this gets even worse with the case of real cells with real probes. So this is no longer solutions in a cuvette. This is cells loaded with nucleotides in this case. We chose nucleotides because they all go to the same locations on the cell. In this case, Belen prepared some I think it was NIH cells loaded with these 4 nucleotides of known lifetimes. Sorry. And these are the recovered distributions of lifetimes. The phasor plot of each of individual images has some weird cloud. This is because you have the bulk of the stuff coming from the nuclei, and then there's some stuff from the background. This is non-specific dyes in the cytoplasm of the cell. So the bright stuff is the nuclei, the cell is much bigger. So only taking into account the pixels within the nuclei, so this cloud here, solve for each individual point in the phasor plot. And as you can see, we're obtaining very nice 3 single modal distributions, which, okay, they're very broad, especially the ones for the short lifetime, but at least the peak position of each of them corresponds to what you're expecting. So I think this result is pretty neat, and this was published in the same publication. We only showed the example, the actual figure we showed is this one. It's basically the same idea. We don't show the distributions, but we show a bar plot. The same, we have loaded with three dyes. In this case, it's acridine orange, nucleus, rose bengal, and we recover the lifetimes and the fractions at each pixel of the image. The exact example is not particularly striking because there is no heterogeneous distribution, and this is the one we showed, but we did not show in the paper the 4-component case, which was it was much more interesting in terms of what you see in the images, because there is some weird heterogeneous distributions. But obviously this is a bit of a mess, so we didn't want to show this because it's hard to tell. For starters, you assign, say, the 3rd component, the yellow one, has a bimodal distribution. This is because in some cases it's actually the 4th component that is being assigned to the 3rd. So maybe you solve for 1 pixel and it tells you that the 4th component has an infinite lifetime with a fraction of 0. So you should be discarding that guy, not contributing it to the distribution, and assigning that 4th longer lifetime component to the 3rd. That is why you get bimodal distributions, and everything is kind of complicated to get anything meaningful information. That's why I'm plotting the mean of the distribution, the mode of the distribution to have several points, and so the peak of the distribution. And well, more or less it maps to what you expect, but it is not very, very clear that it is working 100%. We should probably work deeper into this. The last thing I want to show you is the example, the real example we use this for. So a real application. Our collaborator, Suman Ranjit from Washington, he used to be a lab member, sent us these samples of mice that have been fed with different diets. So you have the low-fat diet, so it's a mouse that you've been feeding it carrots and spinach, and you have the Western diet, it's a mouse that you've been feeding it burgers and beer. I don't know what they were feeding it, but it was basically high-fat content. And as you can see by the intensity images, you can already see that there's a bunch of weird, I mean, it could look completely different. So you will get, I think you're having a talk later on in which they will talk about how to use lifetime to measure the free and bound NADH ratio, which is what you would expect in a healthy subject, so we say. But there was a previous publication in which they showed that in the case of feeding the mice Western diet, you saw this long lifetime species component due to the presence of the fat. So in this case, we use the blind component analysis to find the locations of these positions. So this is the recovered lifetimes of the 3 components of the distributions here, of all the pixels, not only for, this is a single frame, but you have a whole mouse liver section. And you have a distribution of recovered lifetimes, and the mean value we report here, I think it is, I think it's the median we're reporting here, corresponds to these positions of the triangle here. And once we had the recovered values of this lifetimes, we then use it to obtain the recovered fractions for each of the components. And as you can see, the ratio between the free to bound NADH kind of stays the same approximately, but in one case, there's a super strong component of the long lifetime species. We could then map these colorful triangle into the positions to see where the fat is in the actual image. And we did this tiling of the whole liver of the mice. And that's about it. The end.
Thank you, Alex. Beautiful, beautiful talk and very nice work.
a bit intense, I would say, mathematically.
No, it's a beautiful. I really have the chance to discuss with you about this work, and I really appreciate the work that you have been done. We will have time probably for one question. Amit has a question. Out of curiosity for your ND space minimization, do you ever end up getting an hyperbolic nature? Derivative goes to infinite for a derivative during your interaction, and in that case, what can I do? And the second one is, can higher harmonic give you further biological significance, or it is more of a mathematical fitting?
Okay. I think I'm pretty sure we haven't found diverging cases. So we've always converged to a solution. And we are recently moved away from the gradient descent. I talked about the gradient descent because it's kind of it's a conceptually easy to understand that you're looking for the slope in each point. But we is recently using a different algorithm that doesn't even calculate the derivative. So I think what you're doing is it's kind of the Newton approach. It's you modeling locally the slope without actually computing the derivative. So I'm not really sure the details, because we're using this as it's a pre-coded function. But no, we have not encountered any problems in which we have diverging solutions.
Okay.
It is true that also we do limit the scope of the lifetime. So it's not like we tell it lifetime can be from 0 to infinity. We tell it lifetime can be from 0.1 to 30, something like that. Maybe if you don't, it probably does diverge at the edges, yeah.
And regarding
the second question, yeah, that is an interesting question. So yes, higher harmonics you can extract meaningful information, because so we have to go to the first slides at the top. If you noticed, when you do the higher harmonics, the higher harmonics kind of capture, oops, where am I? Capture more detail in the decay curve. So if, say, you have 80 megahertz laser, and basically you're obtaining your decay curve between 0 and 12.5, but you have a very, very rapid decay, which is in the range of picoseconds, the 80 megahertz laser is not optimal. You should really need a higher frequency laser. So in such cases, you would possibly gain benefits of using higher harmonics instead of the lower harmonics to capture the details of the decay. So yes, you can, in the cases where your lifetime is say outside of the nice range for the frequency you're using, the use of higher harmonics can be used to measure precisely lifetimes in your samples.
Indeed, Alex, this is the way Dave explained about the multi-frequencing method with the frequency domain fluorometer, the machine.
Yeah, it's a trick you do often, yeah.
Yeah, Dave, you want to comment?
I just want to comment, Alex. That was one of the best phaser talks I've ever heard. I loved it, and I include my own talks in that when you say it. So terrific. I mean, I think I understand this a lot better now, what you've done. Thank you very much.
I agree with that, Dave.
Thank you. Thank you.
Okay, so we will move to the next talk, and if any of you would like to get into more detail with Alex, we will have the breakout rooms after the 3 sections that we have now. So Suman is here, so you can share your screen, Suman, if you want. Meanwhile, I will introduce Dr. Suman Rashid. Is assistant professor at the Department of Biochemistry and Molecular and Cell Biology at the Georgetown University. I was thinking about Suman, and since I remember when I joined LFD, he was doing this wonderful collaboration with the clinician, and I was really fascinated with this project in which he started to tell me about the use of level 3 and microscopy and nonlinear microscopy in order to understand the fundamental and the utility of this technique for using as a fingerprint for pathologies. He were using, we are talking about autofluorescence and harmonic generation, and he will enlarge about this during the presentation. We enjoy a lot working together. I think we own some records in some of our last paper. I always enjoy one of the last one in which I remember I was trying to write yesterday this, and we were talking about with Enrico. One day we were discussing with Enrico something about the famous Nature Protocol paper, and we came up with the thing that we need to explain something else. And basically, we did all the experiment. We ran the measurement, and then we analyzed all the data, and we wrote the paper within a week. It doesn't mean that the paper got published in another week. It took a little bit longer, but we really enjoy working together. He's a great scientist. He's a close friend, and I'm very happy to have you today here in the workshop, Suman.
Thanks. Can you hear me?
Yeah, perfect.
Yep.
Okay. Let me. All right. So, and you can see the screen, right? All right. So, when I originally planned on giving this talk, it was supposed to be applications of microscopy. But then, after talking to Lionel, I thought it would be a good talk, a slightly different talk based on metabolic optical imaging. So it's going to go a little bit more into the history of the optical imaging, optical metabolic imaging, or metabolic optical imaging. And we'll see like how phasor fits into that category. So of course, Enrico and Dave is there. Alex already talked about some stuff, so I probably won't have to go into too much detail about the phasor. So I'll start with a little bit of historical perspective. And the front picture is where I am. That's my university now, where I work as an assistant professor. And thanks to Lionel for this really nice introduction. Yeah, the paper took less than a week to write, maybe. It took a week to get accepted, but it took 3 months to come out, or 4 months to come out. So that's a different issue. But anyway, going back. So most of the optical imaging, optical metabolic imaging, you can actually get the roots to this paper from Beaton Chance in Science in 1962. And it's looking at intercellular oxidation and reduction states in vivo. And what they're doing is they are looking at mouse, anesthetized mouse in kidney and brain. And this is not a fluorescence imaging experiment. This is 1962. So it's a more of a, it's a spectrophotometer experiment, right? It's with the objective. And what they're looking at is they're looking at what happens to the kidney and the brain under different stimuli. And one of the things they saw, and it's kind of important, is they looked at DPNH, which is basically NADH, it's the old nomenclature. And they looked at between the perfused kidney slice, which is shown in here, and the unperfused kidney slice, there is a change in the spectra. So that there is something happening in the spectral range over there that you can see. And this is all UV excitation. The other important thing that's in the paper is if you change the breathing system, so you're changing from oxygen to nitrogen, and going down here is actually increasing the fluorescence intensity. You see, based on the different gases that you are actually giving the mice, you see this change in fluorescence intensity. So there is also a change in the intensity and spectra based on how you are changing the situation, right? And that sort of started the field, but the imaging started coming along after a 2002 paper from Watoev's lab, where they did 2-photon excitation spectra of the NADH, NADPH, FAD, and LIPDA. So these are the excitation spectra, and you can see around 700 to 750, you can excite NADH. And FAD, on the other hand, has a larger excitation spectrum, but you can also excite FAD at the same wavelength range. And based on the microscopes that we have, based on the lasers that we have, titanium sapphire, this is a proper place to excite. And the emission spectra, you can separate out the NADH emission spectra and the FAD spectra by filters. That sort of started the field. And the question then became, okay, what can we see and what can we do? So a few of the figures that I took for this presentation are from this review from Kyle Quinn in the University of Arkansas. And what we can see is basically this, right? We're looking at, it's a very simplified diagram of metabolism. And you are looking at NADH. You are looking at mitochondria or cytoplasm or outside in the nucleus. And it's two of the components, NADH and FAD, two of the coenzymes, are part of most of these metabolic pathways, TCA cycle, PDAC, glycolysis, lactate dehydrogenase. And if you look carefully, the NADH is brighter and NAD plus is dark. The reverse goes toward FAD. FADH2 is darker. FAD is bright, because these are the fluorescent species you can see. FADH2 and NAD plus are dark, so you cannot see. So the idea is, can we use the NADH and FAD to actually see what is happening in metabolism? And that gave rise to this metabolic optical redox ratio, which is basically an intensity ratio of the FAD by FAD plus NADH. Now I have to tell you that there are different definitions of this. There are different people define it differently. Some people define it FAD by NADH only. Some people define it NADH by FAD. So you have to look at it carefully if you are comparing between paper to paper. But the idea was, okay, if we can do this, and can we actually correlate it with the actual concentration of the NAD plus from NAD plus by NADH, which is known to be a metabolic index. And that basically is possible with 2-photon excitation. If you do 1-photon excitation, you are going to put so much power in the system that it's going to fry the sample. So especially biological system. So the 2-photon excitation lets us do that, and we can look at the lifetime and the quantum yield and the intensity to study metabolic changes. And so this is basically the way what we can see and what we can do. Now, of course, this requires validation. And one of the major validation comes from the intercellular cofactor ratio based on liquid chromatography mass spectroscopy, which has been gold standard in the system. Of course, it doesn't give you cell-to-cell variations or intercellular variation, but this has been biologically the gold standard. Now, one of the papers that talks about it, they looked at human bone marrow-derived mesenchymal stem cells, and these stem cells are going into different types of differentiation. So there's adipogenic, osteogenic, and emissive propagation, and trying to see what happens after the induction, right? So if you look at the optical redox ratio as defined by here, FAD by NADH plus FAD, and if you look at the LCMS ratio, NAD plus by NADH plus NAD, and these three colors are three types of differentiation. That's what they are. You can see a very good correlation between the optical redox ratio and LCMS. This correlation was much less for if we measure the LCMS of FAD by NADH plus FAD. Now, in terms of imaging, and this is the paper, you can look at in emissive propagation, osteogenic and adipogenic, you have very different redox ratio. And you can see the subcellular differentiation here, and come back to that in a second. But also, another paper that looked at it is from Kyle's lab again, which looks at optical redox ratio and in precancerous epithelial tissue. And I'll come back to this precancerous epithelial tissues again. So these are epithelial tissue which has different types of, they're differently modulated because some of them has higher chances of getting cancer. And in this case, the LCMS ratio and the optical redox ratio are highly correlated in both definitions. So there seems to be a correlation between the LCMS measurements that has been done historically and the optical redox ratio. Now, of course, you do not get the intra and intracellular variability that you can get with LC-MS, in any optical imaging. That's why we do most of the imaging, because of this much more detailed information that we can get. Now, another validation is, of course, which is the gold standard now in a way, which is with OCR, oxygen consumption rate, which is basically the rate in Seahorse assays, which is everybody uses. Again, Seahorse assay is a solution measurement right on top of the cells, which measures the not single cell level. It measures whatever happens to the whole system. Now, in this case, again, it's Nirmala Rajaram from University of Wisconsin and her lab, and they have been looking at comparing it with what they call IN-OCR, which is oxygen consumption rate and proton production rate, which is, again, a method of looking at metabolism. And what they did is they blocked the cycle of mitochondrial metabolism by different pathways. So they added oligomycin, which inhibits the ATP synthase. Then they added FCCP, which takes out the uncoupling from NADH, so they uncouple the transport chain. And then rotenone and antimycin, which blocks complex 1 and complex 3, and combined together, they basically block the, change the metabolic pathway. And this is basically what they saw in NADH and FAD. These are just intensity images, but these are redox ratios between 0 to 0.6. But the main thing is here, if you look at the redox ratio, oligomycin decreases the redox ratio, FCCP increases the redox ratio, and antimycin A and rotenone again decreases the redox ratio. It's the same exact behavior that you see in the IN-OCR. So this is another validation that whatever we have been seeing with this particular definition of optical redox ratio, it actually validates very well with IN-OCR. Similarly, they have looked at normoxia, so normal oxygen concentration, and then they looked at what happens if you put them in hypoxia and then reoxygenate it. What happens to the optical redox ratio? So again, this is optical redox ratio. This graph is in OCR, 4 different types of cell line in both in optical redox ratio of 41, 4007, it goes up. FARN, it doesn't, and NR, it actually goes down a little bit. Same exact behavior here. And what they realized that by looking at these, they can actually look at the metastatic potential of the cells, and these cells are more metastatic, so they have a higher change. So this is another sort of another validation for the approach to BOR. Now, this is all intensity-based measurement, right? So it's a ratio of the intensities, whatever definition you want to use. And then we do lifetime experiments. So the question, of course, becomes, why do we do lifetime experiments? So you have to think in a way that intensity of any of the species depends on sum of a fractional intensity, which is the ratio of the quantum yield multiplied by the fraction of that particular species. Now, if it's just one species, FAD and NADH, it works fine. But the problem is, it's not one species. Again, I'm going to go back to this. And if you look at it, there is protein-bound NADH, free NADH, which has very different quantum yield. Free FAD, protein-bound FAD, I couldn't find the number for this, but they also have very different quantum yield. And if you do think about that, then whatever you're seeing in FAD or NADH is going to be completely dependent on what is the fraction of each of these species are present, which makes this correlation sort of difficult to do. And again, in free NADH, the 2 rings up here collapses, which are close proximity, so that decreases the quantum yield, and that's shown is 0.02, at a very low level. And when they are actually further apart after binding to protein, they go away and it increases the quantum yield. So that becomes very important. So we decided, okay, we will do we didn't decide, it's been decided before, lifetime experiments. And lifetime, after all the talks that Alex did and Enrico did earlier, I don't have to say anything. It's just basically the time the molecule spends on an average on the excitation state, and you can get a decay in the old traditional way, and you can find the lifetime. And I'm going to go a little bit, before I go into phasor, I'm going to talk a little bit about just the TCSPC, the time correlated single photon approach, which has been used in the biological system for a while also. But the big thing is this is independent of concentration, so we can actually calculate the lifetimes of these species, and maybe we can correlate that to the optical redox ratio or the metabolism. So the first paper that sort of started that is from Devin Bard and Ramanujam. I don't know how I'm saying that, but they are looking at MCF10A breast cancer cells, and they are looking at how many cells are there. So they're looking at a level of cell density in a sense. Right? And you can see that the lifetime in low cell density to high cell density changes. And what they saw, they fitted this exponential decay by 2 exponentials, and they calculated the free and bound lifetime. But what's more important here is the free to bound protein ratio. So that's the ratio when the number of cells are 25,000, this is 100,000, and this is a million. And you see this really large increase in the free to bound protein ratio over the number of cells. Now, if you go to same system, but you start the NADH, so you're decreasing the metabolic factor, you actually don't see as much of a change. And if you do it with potassium cyanide, which would move it to anaerobic metabolism, you see this system basically behaving similar to serum stuff. So this gave us the way that, okay, maybe looking at lifetime would help us to understand metabolic changes. And again, this is another paper that deals with the precancerous epithelia, which I talked about before. So these are different cancer cells. And here, this row is normal, then these are low grade, and this is high grade, and similar is true. This is the optical redox ratio. The definition is different again here. And you see the redox ratio changing, going down, but also changing depending on how much precancerous cells are. Same thing is here. You see average NADH lifetimes, and this is only average lifetime, is changing this way or this way and this way, right? So by looking at that, they found that the contribution of protein-bound NADH between the normal, low-grade, and high-grade cancers or precancerous epithelia changes. So this is decreasing. And so the main point of the paper is that the increase in free cytosolic NADH that occurs in anaerobic glycolysis not only decreases the optical redox ratio, but also produces a decrease in the mean NADPH. And this is from Melissa Scala when she was a graduate student, I think. And it was published in PNAS in 2007. Now, another paper which looked at it in more detail is, and in a very different organ, is brain. So most of the publication in this field comes from cancer, but this is looking at brain, and basically they are putting topical reagent, and they are looking at what happens to the metabolism. So they have their control, and then they are blocking the glycolysis by 2 different drugs, D-glucose and iodoacetic acid. Then they're blocking the Krebs cycle by, again, 2 different ways. Electron transport chain by rotenone and potassium cyanide, oxidative phosphorylation by FCCP, and they have a focal seizure model because it's looking at actually that. And that's just a map of where different things are getting blocked. But if you look at the average lifetime, and this is again just looking at average lifetime, between the samples where there is glycolysis blocked, it has a longer average lifetime. But if you block the TCA, the lifetime goes away. If you block the electron transport chain, it goes to the lowest. And then if you uncouple them, by using FCCP, it actually goes back to the level here. So that's sort of, again, another validation that this works. A validation compared to OCR from Seahorse assay was shown here. So there's a lot more data here. I'm not going to talk about MCR because it's different. But what you should notice is the free to bound ratio, redox ratio, and OCR. And free to bound ratio here in NADH is measured by lifetime measurement. So if you free to bound NADH ratio goes up here, the way it is defined, redox ratio should go down, OCR to go down. And if you look at the mitochondrial stress test by oligomycin, FCCP, and rotinon, you see oligomycin increases the free to bounded NADH ratio. These two goes down. FCCP, it doesn't change a lot, decreases slightly, these two goes up. Rotinon, this goes up, this goes down. What about glycolysis stress tests? Glucose, oligomycin, 2DG, you look at the glycolysis, and again, if this goes up, this goes down. This goes up, this goes down. So there's a very good correlation between different types of measurement, and you can say that, okay, we can see it. Similar things, and because I haven't introduced phasor and I was not sure how the talks are going before me. So if you look at Chiara's, Stingari, and Carapate, their paper on wind signaling, you can see that exactly similar thing was done with comparing the phasor to all these drugs, and it's in EMO journal, published quite a few years ago now. Now, the lifetime imaging and phasor approach. So you already heard this. So let's say if we have a 256 by 256 image, we have 65,000 points. So fitting each of the points individually is very cumbersome. And that's one of the reasons we do phasor. So this is just looking at a laser pulse and then the decay. And what we do is we transform them by cos and sine. The Fourier transform gives us the phasor plot. And by looking at, if you have a very fast decay, you can see the sine multiplication would be basically 0 and cos multiplication would be higher, close to 1. So it gives you the value up here. But if you have a very slow decay, which basically didn't decay here, you would get, or infinity lifetime in a sense, you will come here and something along the middle would come somewhere in the middle. Now, one of the biggest thing about the phasor is the reciprocity principle. So what I mean by that is you have your fluorescence intensity image and you transform them to the phasor. Now, once you have transformed them, every point of this image gets transformed to a point in the phasor plot. Now, you can use part of this phasor plot to select what kind of lifetime signature or phasor signature they have. And you can see where in the image that goes through. This is very important because let's say if you have a very few pixels of a very different lifetime, and that would be a very separate position here, and then it will allow you to look at that, which conventional fitting approaches would not allow you. The other thing is you can also look at the intensity image, select out a portion, see what their phasor signatures are. And because the reversibility of this whole process is very fast, and you can basically look at biological system and complex biological system much better than a fitting approach would allow you to. The other thing that actually is very important, and Alex has gone through this quite more detail, is the algebra of phasor. So if you have two components, and if you have a point in the image which has both of these components present, the fraction of each of these components will determine where it will be, and it will always be in the line that is joining the two. Now, based on that, you can actually calculate the fraction of, so if you have something which is distributed like this, we can actually change it to a fractional intensity plot, and we'll look at that. So now the procedures, you do the image acquisition, you calibrate the acquisition. The calibration can be done using a fluorescence dye. So let's say this is the position of the fluorescence dye. What we do is, and when the system is not calibrated due to delays and other electronic stuff, it will come, let's say it comes here. This is the original position. So you first correct for the angle, and then you correct for the modulation. So you push it back here. So you know how much of this change you need to do for correcting other sample. You can also use a scattering sample, which has a lifetime of 0. So it will go the reverse way. And once you have calibrated, you can reference all the samples. And then you can do masking, image filtering, and intensity selection by looking at different areas, different intensity areas, and different types of system we have seen. We have looked at biological fluorescence, which I'm going to go in a bit more detail soon, freight, concentration, pH, and you can do different types of analysis. So I don't know if Alex talked about it, but we have gone up to 4 components now. Now, reasons of doing frame is fit compared to other phaser, compared to other. It's fit-free, it's visual in nature, so it's very intuitive. The decays and phasor properties assigned to the molecular species and not lifetime, so that's important. It's an image, so most of the analysis procedure you can use for image analysis actually works on the phasor also. And the other thing is filtering without changing the resolution of the image. So what I mean by that, let's say this is your phasor diagram. You can do a filtering by assigning a median filter and assigning the average phasor to the center of that filter. You can also do a smoothing. So if you do a median filter, you decrease the spread, but it doesn't change the original image. The original image still has the same exact resolution. In comparison, if you do a smoothing filter, original image loses its ability to look at that, right? And then the reciprocity principle, which we talked about a little bit ago, and the linear addition properties of the phasor space. So based on that, how do we do metabolic imaging? We use protein-bound NADH and free NADH. And free NADH has a shorter lifetime, which appears here. Bound NADH has a longer lifetime, appears here. If it's just these two species, the distribution would always appear along this line, which is called metabolic trajectory. And the shift along this trajectory tells you how much, what kind of metabolic changes is happening. The first paper that we was looked at from Enrico's lab was from Kiera, which is looking at neural stem cell differentiation. And if you add reducing conditions, so by potassium, adding potassium cyanide, it goes more towards free NADH. If you add oxidizing conditions, so hydrogen peroxide, it goes more towards bound NADH. By understanding that, they also looked at glycolysis. So if you add high glucose, it goes towards free NADH. And if you use sodium dichloroacetate, which is push it towards oxidative phosphorylation, you see it's more going towards bound NADH. So now we have a way to look at changes between the glycolysis and oxidative phosphorylation, at least the relative fraction of that. So Rupsa, who was a graduate student in Enrico, and this is from Enrico's lab, they looked at in a collaboration in Huntington mice, and they're looking at metabolic reprogramming in astrocytes, and they're looking at striatum and cerebellum. And by doing that, what they did is they looked at what happens with this Huntington disease and how the metabolic imaging NADH changes with Huntington disease. So they looked at Huntington and wild type disease, young mice comparatively 20 weeks and old mice greater than 80 weeks. And if you look at the striatum, you see a huge difference between the wild type and the young Huntington mice. But old mice, there is not much change. This is sort of shown here. In striatum between the wild type and the young, in the Huntington disease, there's a huge change in the bound NADH fraction. However, this is much less in old mice. But in cerebellum, which is resistant to Huntington disease, you don't see these changes. Now, in my own work, I wanted to give a glimpse of it. So this is work with Paulo Sassone-Corsi, who has sadly been passed away, from Center of Epigenetics and Metabolism, and Lorena Aguilar, who is in UNAM in Mexico, and of course, Enrico. And this is looking at spatial dynamics of SAT1. Now, what we are looking at is we are looking at gene expression. So we're looking at gene plasticity and how this works in presence of different writers, readers, and erasers. So the writers modify the either the histone or the DNA. The readers read it and figure out what changes in the gene expression or the plasticity needs to be done. And then the erasers actually takes whatever the writers wrote away, so new things can be, or modification can be made. So SAT1, which is a protein, which is also a deacetylase. It's a clock protein which looks at our sleeping cycles and stuff like that. But it's also a deacetylase, which is an eraser. And we figured that it has to bind to this for its eraser activity, which means its diffusion would change based on the activity. So we can look at that SAT1 activity by diffusion. And to do that, we used a technique that Enrico, me, and Luca Lanzano have done before. So this is just simulation of beads moving very fast, medium, and slow. Now it's 256 by 256 point. If you want to get a individual decay curve, you have to, or fluorescence correlation spectroscopy at each point is going to take 10 seconds at each point. So that's not even possible. So what we did, we looked at very short intensity decays, and we looked at, and we converted them after we calculate the autocorrelation function of the short intensity decays, we converted them to phasor plot actually, and we saw that. So this is sort of what you would expect in single exponential, but this is what you see when you do a very short lifetime. So this is 64 multiplied by 20 microsecond. That's short of a lifetime. And then you can transform them, and you can use the phasor to select out slow and fast diffusion based on the color. So slow would be up here, fast would be up here. Now, we can also look at what happens to the NAD plus metabolism in compartmentalized nucleus. So this is nucleus, and a salvage cycle, which is the part of nuclear cycle, involves NAD, NAM, and NMN, and different enzymes. And we wanted to see what happens, and if we can see this, these changes here. So we, of course, look at phasor film of NADH to look at the nucleus. So going into the actual expectation, idea was we would modify the salvage pathway, and SAT1 would become active, and we should see that. But when they are active, we would expect the SAT1 slow SAT1 diffusion, and when they're inactive, we would see a fast SAT1 diffusion. We will also see a higher ratio of free NADH when SAT1 is active, and lower ratio of free NADH when it's non-active. And so to do that, what we did is we took the salvage cycle and blocked the SAT1 activity by mutation or drugs. We also added FK866, which is a drug that blocks this enzyme, which is the rate limiting step of this process. So these are our direct control. But in our metabolic control, we added NAD and NAM and see what happens to this. So to show the data, so this is basically the film data from the nucleus. So you see the images, then they are marked for only the nucleus, and then they are looked at the lifetime. And between wild type and SAT1, you see there is a change, the SAT1 negative cells, so inactive cells. So this blue is the wild type, red is the SAT1 negative. It goes more towards bound NADH. However, if we add EX527, which would block the SAT1 activity, we see slowly it's going away the similar manner as the SAT1 negative cell. However, if we add it to SAT1 negative cells, which shouldn't matter, it doesn't. We don't see any changes. And SAT6 negative cells, which is not in nucleus, which is not a part of this, there is no change compared to the wild type. Now, what happens to our diffusion? So slower diffusion is going to be shown in the purple, and higher diffusion coefficients are going to be shown in the more red and orange. And you see the SAT1, these cells, which is SAT1 is tagged with EGFP, but they're positive. You see this purple flecks all over. However, if you make the SAT1 negative, make it inactive version of it, it goes away. So, this fraction of this slow, so the bound fraction, which is actually active, is going away. Similarly, if we do the same thing with the drug, as a function of the drug, how much time the drug has to interact, it starts going away again. So this was our direct control experiments, which correlated our film data with our diffusion data. Now we went back and looked at the indirect inactivation. And we did it by blocking again NMPT. We added NAM, which is actually inhibitor of SAT1, and by adding NAD+. So in non-treated NAD+ and NAD over time, you see there is a change, but then the change slowly goes away, which is shown here. However, in NAM, you see the reverse change, which you expect from this, right? And in SAT1 negative cells, there is no change. However, if we do FK866, you see basically similar value as adding NAM, it's going away, but SAT1 negative cells has non-significant changes. We looked at similar things with diffusion. So Again, we add FK866 over time, it stops the SAT1 being active. So by blocking the salvage pathway, we are making SAT1 inactive. On the other hand, we take these inactive cells and we treat them with NAD plus or beta NMN, and you see the activity starts coming back. So this was sort of a first direct experiment where we looked at how metabolic changes affects the activity of a deacetylase, and on the reverse, activity of the deacetylase changes the activity of, or shows a change in the metabolism. So that got published a few years ago, but there is a word of caution, and this is sort of the papers that Lionel was talking about before we started. So if you look at the publications on Phaser, you see different trajectories for NADH, different positions for bound NADH. Some people put it here, some people put it in the middle. Why? Well, there's one of that comes from the difference in the bound NADH lifetime, because depending on what protein it is binding to, it can have different lifetime, and that would change the position here. But a bigger problem seems to be how this data is analyzed. And part of that is how this data is transformed. So I'll come back to this in the next slide again. So basically, most of the people takes data at on a Baker and Hickel card based on TCSPC approaches with a gain of 5, which is about less than 10 nanoseconds. So it's about 8 nanoseconds. Now we are trying to take this and we are trying to fit the decay that appears here, which didn't decay to 0. In that case, the fitting doesn't work very well. Alternatively, if you're trying to transform this to a phasor, this is a non-repeating signal. So Fourier transform is a repeating signal. So that doesn't work very well. So what Enrico proposes, and we showed in the paper, is you should, if you're trying to do a phasor transform, you should take a much lower gain, so gain of 2, which is 25 nanoseconds. You select a 12.5 nanosecond window out of that, which is actually repeating pattern, and you could convert that. That's the proper transformation. And so these sort of work got Lionel and I published in 2 continuous papers. One is to look at bound and free NADH, the other one is the proper transformation. And because these differences, you would have different metabolic trajectories from different papers. So it's worth looking at it with a sort of a cautious mind. Now, this effect of proper transformation can be seen very easily. So this is Flimbox, which is frequency domain, which we use often. If you do Baker and Hickel gain of 2, you basically get the exact same results that you get in a Flimbox. However, if you do the gain of 5, you see a very different trajectory, and it even goes outside universal cycle. And this is sort of what we saw this is just a combination of these 3 together. In terms of actual cells, if you do Flimbox or Baker-Hickel gain of 2, we basically get similar distribution. However, if you do gain of 5, we get a very different distribution. So if you're comparing this with this, you're not going to get, the relative fraction might work, but the actual quantitative information will not be around. So what about fixation? So it has been shown that MDA, so this is a triple negative breast cancer cell, MCF10A is a non-cancerous epithelial cell. And if you look at the live and fixed sample, you basically see the same behavior, different extent, however, similar behavior in lifetime of NADH. This doesn't work for FAD. FAD completely changes, but in NADH it works. So this is again control sodium cyanide to deoxyglucose coming down, going up. Control sodium cyanide to deoxyglucose in live cell coming down and going up. How about each pixel to pixel? So you can take the same area of the live and fixed, and if you look at a co-localization histogram, you see a very high degree of co-localization. It seems to say the exact same thing that is present in the live cell shows up in the fixed sample also. In phasor, yes, it get transformed. Of course, the lifetimes are different, so there's a shift. However, it's still along the metabolic trajectory, so we can compare between similar preparations. And finally, if we look at mean lifetime distribution on normal pancreas and cancerous pancreas, it will shift to the lower lifetime, and that's exactly what they have seen. Now, in a few minutes, I'm not sure how much I have, but I'm gonna just introduce this. What else can we see? We can see oxidative stress. So this is Rupsa's work, and it's basically transmission and these are transmission and fluorescence images. She was dealing with cells which is fed with oleic acid, and she saw this different behavior which is different than the metabolic trajectory. So you can just color code them. However, when she did coherent anti-Raman spectra or third harmonic generation, which looks at lipid droplets, you can see these signals are co-localized within the lipid droplets, this signal. And this is coincident, as shown by Raman, with the triglyceride peak. Now, in terms of oxidative stress in human pluripotent stem cells, so this is normoxia, and this is hypoxia going along the way. And as you can see, this is there's a change here. So this is just the NADH. I don't have time to go into it. But in terms of oxidative stress, you start seeing that, okay, with more and more hypoxia, the more and more this LLS species is presenting. You can see it in the phasor plot, you can see it's the quantification. But of course, this is sort of a broad quantification. So in vivo, and I wanted to work a bit more. So we first created this, that was a few years ago, maybe last year, fraction intensity contribution in a 3-component system. So the idea is, if we have only 2 components, this would be along this line. However, if we have a third component, based on how much this third component is, it's going to drag it towards that component. Now, it would always be within these 3 points. We looked at that. What we did is but if you can think about, you can select a point inside this, you can project a line from here to the original metabolic trajectory, and you can do that for the whole distribution, and you can transform it to a distribution. So by doing that, we can calculate what's the fraction of this component or what the fraction of these components. So by that, and then based on that, we looked at this high fat, high cholesterol Western diet and normal diet back in some years ago. And this is normal diet, but with the fat diet, you see the changes in the liver. These are all fat droplets. But we can do the quantification, and we can see how much is this long lifetime species is present. This is work with Alex. I'm not sure has time to go through, but this is basically, we converted that 3-component to now 4 components. So these are 4 different dyes mixed in, and you can see they are always along different lines, and we can actually calculate their fractions. We have looked at cells, which has 3 different nuclear dyes again, and we can, individual cells, we can calculate their fractions. They're only going to be 1. However, when they're mixed in different amounts, we can get those fractions. This seems to be really interesting to use in large biological system, where every small area of the image gives you a different value. So we have a much higher statistics to get into. This is blind resolution. Alex already talked about that, so I'm gonna skip that in the interest of time. But again, we basically were able to get the numbers that we have done previously with knowing what the components are, but in this case, without knowing any of the components. Finally, to acknowledge LFD, where I did almost all this work. Some of the work continued after I moved to Georgetown as my independent position. Enrico, of course, nothing would have been done without him. All of this was his idea, of course. And whenever we come to him, he says, we can do it, don't worry. Lionel, I don't know how many projects we have worked on so far, and hopefully that continues. And this is just because he loves Ramones. And then Alex, Belen, and who are partners in crime in the last couple of papers, and then Sasha, who we have been working together since I joined with the diver, and I'll talk about it in the next time in the diver talk. People in UCI who have been great collaborations, people in Georgetown, Moshe Levy, who has been a long-time collaborations and his lab. There are other projects that I didn't have, of course, time to go through, and none of them are published also. But there's a lot of work on cancer that I'm doing using the techniques that I learned in LFD. And then, of course, at the end, but not the list at that time. Anyway, as Dave for all the help throughout the year, telling me. And yeah, finally again to Enrico, because without him, none of this would have happened. And funding sources and just stuff. And that's it.
Thank you very much, Suman. Great talk. As you say at the beginning of your talk, we were discussing when we were organizing the course, and I say, Suman, you should give this talk, not me. And I was right. So I think you give a beautiful coverage of all the metabolism. And if there is a question in the audience, I think we have time for one question. If not, then all the people who would like to discuss with you, we will have these breakout rooms. So just a second. I'm looking at the chat now. Or if somebody wants to raise the hand, it's also possible. We will allow you to ask the question. Suman, I do have a question. You know, regarding to the liver, when you show this data that you have that you did here is you analyzed this using the previous analysis of the cursors, right? I didn't remember if you used this analysis with the 3-component, and on top of that, is you ever uses these algorithms based on the minimization of the spectra in order to split groups. I found interesting if you have this information, if you ever test in order to move basically all these large samples, which are almost impossible to analyze tile by tile, and you have to use some artificial intelligence or something like that for do this analysis.
So the spectra part, which I haven't talked about here, but that is actually very easy to do here because the change in the lifetime signature or phasor signature is so much. It's very easy to do. I haven't done that, but the data would work for sure. The other part about the 3-component analysis, yes, we have we did that. Originally, it was developed to look at that only, the 3-component analysis. So that's in a different paper. But then the actual theory of it got lost in that paper, which was a very biological paper. So Enrico and I decided to actually work on that theory and publish it separately. And then Alex basically did Alex and Belen, they did similar experiment on similar systems to show that what we saw actually works on that sample also. So it was, altogether seems to be consistent. The thing is, if you don't have phasor, you will never see that lifetime because it's 10 nanosecond, 8 or 10 nanosecond. So by getting an 8 nanosecond window, it's basically flat. So you would never say that. So that's the other thing that I didn't put in why I use phasor, and I hope Enrico liked that idea that why we use phasor.
I think this was the original discussion for which we did the first paper of the 2 papers about how to collect the entire period and how to transform the data.
Yeah.
Okay, Suman, thank you very much for your talk. We will move on to the last talk of this row. The next talk will be given by Nick Hede. I already introduced Nick Hede yesterday, but I will again say that he's project scientist at the Department of Pharmaceutical Science, University of California, Irvine, as well as a member of the Laboratory for Fluorescence Dynamics. And today, Nick is going to talk about a particular application of the cyan cosine filter for spectral imaging, but using a light sheet microscope, which is, I think, really unique. And it opened many possibilities for different application. I can guess that one of the most is for developmental biology. And we did some experiment in this way, and I think he will illustrate us with all this application, Nick. Now I think you are able to share the screen.
Yeah, thank you, Lionel, for the introduction. Let me try to share. Okay. Okay.
Yeah, right.
Is this the full screen view for you?
Yeah, yeah.
Okay. Okay, so yeah, welcome everybody, and thanks again, Lionel, for having me. So this talk is about doing hyperspectral imaging on the light sheet microscope using phasers. So in the first part of the talk, I will give you a brief introduction on light sheet microscopy. I think Lionel is going to give a presentation tomorrow to discuss the light sheet microscope in more detail. And then later in the second part of the talk, I'll show you how we can do hyperspectral imaging with this kind of setup, which is not straightforward using conventional methods. Okay, so first, I want to give you an overview of the different principles of fluorescence microscopy. You can generally split the methods in two categories, although of course there are hybrid methods that take elements of the two and recombine them. But in general, generally speaking, there is wide field or camera-based detection, and there is single point or laser scanning microscopy. So for the camera-based detection, you are illuminating the entire field of view with a beam of light, and you're detecting from the entire field of view. In the simplest case, that could be just using your eyes and looking through the eyepiece. And in this case, the array detector would be your retina. If you want to store and quantify the images and do high-speed recordings, you can use a camera, a CCD camera, or more recently, CMOS camera. And the advantage is that it's very fast because you can just snap an image within a fraction of a second, a couple of milliseconds. And this technology has really evolved in the last decade. Now all of you probably have a phone that has a camera with, I don't know, 8 megapixel, 12 megapixel, 20 megapixel. And that was something that was not possible just maybe 10, 20 years ago. Yeah, so these cameras also in the scientific, for scientific applications have evolved very much. A problem with this approach, if you just illuminate in this kind of epi configuration, meaning just yeah, dumping light on the sample, is that you will also excite fluorophores in planes that are above and below the focal plane of your objective. And these will give you background. So if you're using this kind of epifluorescence wide field microscope and you have a thick sample, the images will look kind of hazy. And this has nothing to do with the resolution. It's just that you don't have optical sectioning, so you get all this background from other Z-planes. Or, yeah. Then on the other hand, we have those single point detection or scanning methods. This is your confocal microscope or your two-photon excitation system. And the idea here is that we take a beam of light and we focus it using the objective lens into a single point. That point is, of course, diffraction limited. So with a high numerical aperture lens, it's maybe 200-300 nanometers, which defines the resolution of the image. Of course, there are super resolution methods now where you can improve on that, and there will also be talks about that. But anyway, so you focus the light into this tiny spot, and then you scan this spot across the sample. You can either move the sample with a scanning stage, or you can use a mirror behind the objective to move the laser beam. And then you scan the sample point by point and reconstruct the image after detection with your photomultiplier or similar detector in the computer. Now, the advantage with this approach is that before detecting, you can put an aperture, a pinhole, and with this pinhole, you can reject light that comes from areas that are not in the focal point. So from planes that are below or above the focus that still get excited from this cone of light, you can block these photons using a pinhole. Or you can use multi-photon processes which have a nonlinear relationship with the excitation intensity, and thereby you only excite a very tiny 3D volume in your sample. So this gives you very nice optical sectioning, and you can do very clear 3D imaging also in thicker samples. However, because you have to scan your sample point by point, this method is inherently slow. If you are working with a confocal microscope, you see how the image updates, and it's generally in the range of maybe one frame per second, compared to the camera where you easily can get 100 frames per second. So about 2 orders of magnitude difference in terms of speed. So ideally, we want to be fast, like in the epifluorescence, but we also want to have the optical sectioning. So for this, people came up with the selective or single plane illumination microscope, or it's also called a light sheet microscope. And the idea for this setup is that we use split up the path for detection and excitation, often by using 2 objective lenses. So one for detection, and then another typically at a 90-degree angle that creates a sheet of light that illuminates the focal plane of the detection lens. And because it's a sheet, it only illuminates that plane, and we don't generate any out of plane fluorescence, meaning that we don't need any apertures, any pinholes to disregard these other components. So with the light sheet, the advantages are we get excellent optical sectioning capabilities similar to a confocal microscope, about 1 micron sections, or maybe even better depending on your setup. Because we're using a camera, and these can be these large area sensors that we now have, maybe 2000 by 2000 pixel. So we can really have a large field of view at high resolution. With the setup, we have a high light throughput. We don't need any dichroic mirrors to split out the fluorescence light from the excitation. As mentioned earlier, we have this very high temporal resolution to acquire images at rates of 100 frames per second or faster. And another fundamental advantage of the principle is the low phototoxicity, because in the confocal microscope, we are still illuminating the entire sample in Z direction. We are just blocking the fluorescence from other planes. But for every section that we take, the entire Z gets exposed, and therefore we have the potential to induce damage or bleach the fluorophores in the other planes as well. With the light sheet, we only excite the plane that we are imaging, not any planes above or below. indicated here with only what you observe is what is exposed, and therefore we can get inherently lower phototoxicity and photobleaching. One thing I wanted to point out is that I have a couple of papers referenced down here on the left corner. And if you look at the first paper that came out in 1903, so more than 100 years ago. So you could ask the question, well, if the technique is so old, why has it taken us so long to take advantage of this principle? Because if you look at the next papers, the next one is 1993, and then the method really took off with the paper here in 2004. And the reason is that when they first implemented this in 1903, they had no cameras, no electronic cameras where you can record, quantify the images. They had no computers to analyze the data. There were no laser sources. The laser had not been invented yet. So all these factors made it hard. You could only basically look by eye, and then you cannot deal with all this 3D information. So it really took all these other developments to make it as powerful of a technology as it is today. Okay, so one problem with this method, or difficulty with this method, is now we have these two lenses. So how do we accommodate our sample? And there are really a lot of papers with a lot of different approaches trying to tackle this problem. I'm not asking you to read all, look at all these figures, but I just wanted to flash the slide to give you an idea of how many techniques there are out there right now. And this is by no means a comprehensive list. Of course, we took our own spin on the light sheet microscope, which we call the side spin. And this is based on an inverted, conventional inverted microscope, which you can see here on the left. So this is our inverted microscope body with the stage and everything. And then here on the right-hand side, we have the components for to generate the light sheet. So we have a visible light beam that is cleaned up, and then we have a beam scanner, because there are two ways you can generate the light sheet. One is using some kind of cylindrical optics. The other way is that we use a low numerical aperture lens to generate a beam that we then scan in one direction across the focal plane of the detection lens to generate the light sheet. So we use the second approach, using the scanning to generate this virtual sheet, because it's more flexible. You can change the area that's illuminated and so on. So we have these Galvo scanners here, and then we have the typical scanning configuration of the scan and tube lens to generate a sheet. You can also use 2-photon excitation, which is important for thicker tissue samples, and also for the hyperspectral imaging, as you'll see later. Okay, how does the sample handling look like? So we have this custom sample chamber, which has two glass windows. One is on the bottom, so for interfacing the detection lens. And then the other one at a 90-degree angle on the side to interface with the excitation lens. And on the right-hand side, you can see a picture where this chamber is into the, put into the microscope, and we have the detection objective on the bottom and the excitation on the side. Okay, one property I wanted to mention is that in this kind of configuration, you have to make sure that the refractive index is uniform to get an undistorted image. Here on the top left is a schematic of the chamber. So we have the cover glass on the bottom, the cover glass on the side. We have the detection objective coming from the bottom and the excitation lens coming from the side. And if we have a sample that's, for example, an agarose gel or a gelatin or something, and in this case, we just immersed 100 nanometer beads, you don't have any problem imaging this in 3D because the refractive index is the same everywhere. So that works out pretty nicely, as you can see on from the images on the right-hand side. With this kind of objective configuration, we were using NA1 in detection and NA.3 in excitation. We got an XY resolution of about 370 microns, 370 nanometers, sorry, and an axial resolution of about 1 micron. If you have a sample that needs to be supported in some way, if you cannot suspend it in a gel, well, you have to create a support and you have to be careful that it matches the refractive index of whatever is above, which usually is some kind of water-like substance, some cell medium or something with a refractive index of about 1.3 to 1.35, something in that range. So if we choose a support that matches the index, we can also image samples that are on some kind of interface with similar resolution. As an example, I'm showing you the giant unilamellar vesicles. So here we had this resin surface matching the refractive index of water. Then we immobilized GUVs on the surface via streptavidin biotin binding. And then these GUVs were fluorescently labeled. And here in the left picture, you can see a 3D rendering of a GUV just made from DOPC, labeled with Texas red. And then in the middle, we have a GUV which is made from 3 different lipids, DOPC, DPPC, and cholesterol. And this leads to phase separation. And these phases have different affinities for the dye. So we see this contrast and we can see those rafts or islands, and they are moving, of course, because it's fluid. And now with the speed of the spin, we can record movies in 3D. So for example, here we did a recording of 1.25 stacks per second. So basically you can get 3D images at the same rate as you can get 2D images on the laser scanning, which is very nice. And then here's just showing you that, for example, now you could track the diffusion of those structures in 3D as a function of time. Okay, so just again here a little sketch overview of the simplified sketch of the system. So we have various laser sources. We have these scanning mirrors to generate the light sheet, and then we have our cells or tissues in the matching refractive index matching environment, and then we have the camera and detection, and we can choose different emission filters. Okay, so one question that we had is, can you do spectral imaging with the system? You might have used a hyperspectral confocal microscope, and the way it works is that in the detection you use some kind of dispersive element like a prism or grating to split up your fluorescence according to wavelength, and then you have a multi-PMT array and have maybe, let's say, split up the light in 16 or 32 channels. Where you can get this hyperspectral information. This is a problem with the light sheets, as you can see here. So what people have tried is, well, you have this kind of, you have a 2D image. We are working with a camera. And then now in addition, you have this, you have to split the light according to wavelength, which gives you this kind of 3D hypercube for spectral information. So what people have done is they only illuminate a line instead of the entire field of view, and then use the second dimension of the camera to spectrally split the light. And then they spatially scan in the other direction to reconstruct the 2D image. And of course, this takes time, so you're losing a bit of the advantage of the light sheet microscope. I mean, it's still faster than the laser scanning confocal, but not as fast as it usually is. Instead of scanning spatially, you can also scan spectrally. You can, for example, use a tunable filter to change the band of detection. But again, you have the speed disadvantage because it takes time to scan through all the wavelength ranges. So what we want ideally is a snapshot or non-scanning method. So we want to have all the information in one shot. People have also done that. And what they have done is they split the field of view into smaller sub-images, and each of those sub-images gets filtered with a different wavelength. So you get all the wavelengths at the same time. But of course, if you split your main field of view in tinier images, you shrink down your field of view. But with this approach, at least you can maintain the high imaging speed of the light sheet microscope. Okay, so the presentation, the previous presentations, you have already heard about phasers and about using phasers for fluorescence lifetime. You can also do phasor analysis for hyperspectral imaging. And the way this is typically done, again, if you're using your laser scanning hyperspectral microscope, we detect the light from a pixel, from a point, we split it up into the wavelength components, and then basically we record a spectrum in every pixel, and then you have as many spectra as you have pixel in the end. This is a problem for the analysis, because now what do you do with all those spectra? A small image of 256 by 256 pixel would have more than 65,000 spectra. So how do you analyze, how do you deal with all this data? Again, here the phaser is a very powerful method and very easy to use to visualize and quantify the data. So we apply the spectral phasor transform to end up with our G and S coordinates, just like for the lifetime, except that for a lifetime decay, we now have this spectrum that we transform into frequency space. And then we end up with a phasor plot that looks like this one here on the bottom. And the way you read this plot is that the position on the plot is determined by the center of mass of the spectrum. In this case, our detection range is 400 to 700 nanometers, and the position will change, the angular position will change counterclockwise on this plot, depending where the center of mass is. Then the other coordinate, which is the distance from the center, is defined by the width of the emission spectrum. If you have something that is very sharp, like a laser beam, that would appear here on the outer circumference of the circle. If you have something that's completely flat, that's like a white light source, then that would localize to the center of the plot. And shown here, these tiny little distributions are measurements using this phasor method of dye solutions of different colors, ranging from violet to blue to green yellow, orange, and then red finally. So you can see they all have a very distinctive position, and you can clearly distinguish them. Another advantage of the phasor analysis is the linear combination. Just like in the lifetime, if you have two components, for example, here we were mixing a green dye, a rhodamine 110, with a red dye with an Atto 590 at different proportions, and then all those combinations localize along this connecting line. And from their position, you can calculate the relative fractions. So this is another advantage of the phasor analysis. Okay, so now the question comes up, how can we use this principle with our light sheet microscope? Well, here we're kind of doing a lot of steps. So we're acquiring the spectrum in each pixel. Then each pixel we transform to the phasor plot using these formulas in a computer. So why not do the phasor transformation directly in an optical manner? So if we would have optical filters that are shaped in like a sine and cosine function that represent here the phasor transformation, then we could directly acquire the data in the phasor space, and we would not need any of those hyperspectral detectors, splitting devices, and also don't need to do the transformation computationally, although that's not a big problem. Yeah, so we would end up with the same result, and actually the data I showed you was actually acquired with the filters instead of the conventional splitting method followed by the phasor transform. Yeah, just to give you an idea, so these were the custom interference filters that we had made for the light sheet microscope. So they resemble pretty well the shape of the sine cosine function. Then we added a bandpass 400 to 700 nanometers to make sure we block light from regions of the spectrum that are outside this response function. So on the right-hand side, we get the combined optical response of our system, which is still pretty good. Okay, so the first thing we tried is our trusty fluorescent beads. So we mixed 5 different species of 1 micron beads of different colors. Here on the top left, you can see their emissions. And then we took, we embedded them in agarose gel and took 3D images on the light sheet microscope with the sine filter, with the cosine filter, and then another one with the without filters to get the total intensity which we need for normalization. So from those 3 images, we got the phasor information, the phasor space information. And here you can see the distributions of this 3D data set, or I should say 4D, because it's now including the spectral information. And you can separate out all those colors very nicely. On the bottom left, you can see the 3D reconstruction that was labeled according to the selection on the phasor plot. And on the bottom right, you see the phase histogram. So just looking at the angle here on this plot, the distribution of all the pixels, and you can clearly see those 5 distributions corresponding to the 5 fluorescent bead species. We also wanted to confirm that we get a result that is comparable with the conventional phasor-based hyperspectral imaging. So we also recorded one on our Zeiss LSM 880 that has a hyperspectral detector. On top, you see the phasor distributions from the side spin, and in this case, we just made samples which only had those 1 component, so we can be sure that we get the localizations right. And from the plots here on the bottom, you can see we have the center of mass wavelength of the 32-channel detector plotted against the sine-cosine method, and that's pretty much perfect. And then also the modulation, so the distance from the center, the coordinates plotted against each other for the two systems. And again, we get a very good match. You can see that if you look at those pixel distributions, the phasor plot clouds, we have many more pixels for the SPIM than for the light sheet, for the light sheet microscope than for the laser scanning. And again, the reason is the speed difference. In the same time that we took a 3D data set on the light sheet, we were only able to do one 2D image on the laser scanning. This is why we have much fewer pixels here. Okay, so after this verification, we went on to do cells. So here are NIH-DT3 cells on a collagen substrate to match the refractive index. And we stained those cells with 4 different organelle stains, the nucleus with nuc blue, the Golgi with the body pie, and then mitochondria with mitotracker and lysosomes with lysotracker. And here on the top left, you can see the spectra of them. And you can see that they strongly overlap, especially the blue and the green and the orange and the red. So we took a set of images using conventional bandpass filters, one for the blue. So, okay, we can see the nucleus, one for the green region or green-orange. But this already, you see, we get a mixing of the Golgi and mitochondria. And then we have another channel which is supposed to select for the lysosomes, but again, we get mixing in with the signal from the mitochondria. So this, we cannot basically clearly distinguish just using bandpass filters. So then we did the imaging with the sine-cosine filters. You see the phasor plot. And again, we selected regions on the phasor plot according to the emission spectra that you can see on the top, and then painted the pixels in those 3D data sets. And you can see that we clearly get, can separate out the nucleus from the Golgi. You can see, this is a 3D. Unfortunately, I cannot rotate it, but if you would open it on your computer, you could rotate it and would see that the Golgi wraps around the nucleus. And then we have also clear separation of the mitochondria and the lysosomes, which is very nice. Because the light sheet is fast, we can also do hyperspectral imaging as a function of time. So get a 5D data set, so the 3 spatial dimensions, the spectral dimension and the temporal dimension. And you can, for example, do tracking of the lysosomes and simultaneously look at the behavior of all the other structures. So then we went on to a more complicated system, which is the zebrafish. It's a very popular system to study developmental processes. And light sheet microscopy is used a lot on those kinds of samples because you can rapidly get the 3D information. And the zebrafish has the advantage that it's optically clear mostly. So you can usually get very nice, clear and crisp images. So we use this so-called spectrum of fates line to study the developing zebrafish retina. And the idea is that you have different genes and they are tagged with different proteins. For example, a red fluorescent one, a green fluorescent one, a yellow and cyan fluorescent one. And then they get expressed in the cells, but at different proportions, depending on what the cell differentiates into. So here you can see the individual images of this publication where the different fluorophores highlight different structures of the cross-section of the retina of the eye. So for example, the photoreceptors are here on top, which are a combination of red, of the RFP and also the CFP. Okay, so Lionel and Andres, his friend and collaborator, were working on those fish and sent them over, and I imaged them on the light sheet microscope equipped with those sine cosine filters. Here on the top left, you can see kind of sliced off 3D top view of the zebrafish eye. This is just the intensity image. Then in the middle, we have the resulting phasor plot with the pixel distribution of this 3D data set. And then on the right-hand side, you can see the pseudo-colored image where we applied a color code corresponding to this phasor selection here. So in the phasor plot, we see marked by a green dot is the theoretical position of the spectrum of the GFP, the cyan, the theoretical position of the spectrum of the CFP, here down here, the RFP, and then we, as a fourth component, we use the nuclear stain, the Cytox orange. Yeah, but you can see they all combine as we expect because they're expressed in the same structures, but at different proportions. So now this is perfect for analyzing it with these linear combinations. So for a simplified analysis, we chose a color code ranging from cyan to magenta for the linear combination of the CFP and RFP, and then yellow-green for the linear combination of the GFP and Cytox, and this is what is applied to the images here. Then we, if we zoom in into the data stack, into this smaller cross-section, you can see that we get the distributions of the fluorophores that match pretty well what you see in this publication here before. And then we also took a cross-section to resolve in detail, and yeah, again, it matches the architecture that we expect of the eye. So, for example, again, for the photoreceptor here, we have a combination of the cyan and red, but no green. And you can see also this in the profile cross-section. Now, because this is phasors, we can also do more detailed studies. And one nice property of the phasor is, well, you can go to the phasor plot and select pixels on the phasor plot and highlight them in the image. But you can also go to the image and select the region and then reconstruct the phasors or just show the phasor plot from that region. So this is the reciprocal principle. So here we, according to the intensity distribution, we identified these different regions of the retina, M1 to M5, and constructed the phasor plots for all of them. And then you can analyze more complicated linear combinations and color code them. So we can see a more fine layering here of the structures. In this case, the colors not necessarily represent the actual color of the fluorophore, but it's just highlighting different components, different structures that have different ratios of those fluorophores. And you can see we can identify single cells that have is a particular type of cell that has a unique spectral fingerprint, I would say. Finally, this was all with labels, external. Yeah, we had to label all those cells and tissues. So one advantage is if you can do autofluorescence. So in tissues, for example, it can be difficult to get those labels into the cells. And we tried this. We used mouse, ex vivo mouse tissue, intestinal crypts, and they are unlabeled. And we just used 2-photon excitation at 740 nanometer to excite. And with that wavelength, you typically excite NADH and FAD Those are two metabolates that occur in the cells and tissues at relatively high concentrations. So on the left, again, you see the reconstructed 3D intensity image. In the center, we have the spectral phasor plot. And then we can define an optical redox ratio because depending if we get fluorescence from NADH, this is more blue, or from FAD, which is more green-red, that gives us an indication of the kind of metabolism that's going on. So if you look at the bottom left, for example, for oxidative phosphorylation, this leads to the formation of FAD. And this is typically what happens in differentiated cells. And there's also glycolysis, where you have the ratio tends to be more shifted more towards NADH. And this, in our model here, happens, for example, in the stem cell area of this intestinal crypt. Yeah, so according to the definition of this optical redox ratio, we applied this color code along the trajectory, and you can see the color coded 4D data set on the top right. And if we quantify this, so we can calculate the average redox ratio for each slice along X, Y, and Z. You can see in X and Y, there's not much of a change. But in Z, we start with a higher redox ratio, and which then goes down the further we go along the Z axis. And this exactly recapitulates this gradient of the intestinal crypt with stem cells undergoing preferentially glycolysis to the lumen, with the differentiated cells primarily undergoing oxidative phosphorylation. Okay, with this, I would like to thank, of course, Enrico and the LFD, and the people in particular who contributed to this project, including Rachel, Lionel, and Andres. We also got some help from Sasha, who is using those types of filters in his diver instrument, or a variation of those filters in the diver instrument. And of course, at UCI, the labs of Marion Waterman and Tom Schilling for providing mouse tissue samples and help with the zebrafish. And of course, the Institut Pasteur Montevideo that prepared the sofa fish line and shipped all those samples over here. Yeah, thank you, everybody. Questions?
Thank you very much, Nick. Beautiful work. I really enjoy doing this work with you. Dave, have a question. Okay.
Yes. So Nick, a great talk as usual. As you know, Lionel's done work on multi-dimensional phasers linking lifetime data and spectral. It would seem like those zebrafish would be a nice system to try that. Has there been any effort to link those two, and if so, what happens?
Yeah, we haven't done it yet, but we have plans, because you're right. Another advantage of those filters is so we don't really care what the detectors is, because we get the hyperspectral information with the filter. So in principle, we could put a camera there that's modulated, where we can also record the lifetime information.
Because I would expect those different fluorescent proteins, it may not be big, but they're going to have differences in lifetimes too, I'm sure.
Yeah, I think so too. And the other point is that with the metabolism, I mean, this work, I would say, started more, or many people do it via lifetime, looking just at the NADH, where you have the free and bound, for example. So if we can combine all of these, that could be
that would be great.
Yeah, maybe we can have time for one little short question before to the breakout room section. Indeed, if there are people who would like to join Nick, Suman, and Alex to the breakout room session, please place your request in the chat. Also, if you want to join with Enrico, me, and Dave, we can also split in the groups and generate breakthrough session for all of us. If not, we will basically recombine 5 minutes to 16:10. So 16:15, sorry, for the last section, which is the application talk. And the first talk will be given by Susana Sanchez, and then there will be 2 talks by me, and this will be the end of the day. So, okay, for Alex, there is one. There is still not request for the others. You can request over the time. We will be here paying attention because we can assign you meanwhile the break group session is on. So if there is no more question, we will start the break group session. We will recombine 5 minutes to the next thing and continue with the meeting.
So how does this work?
You will just wait. Okay. Just wait, and something will appear that says "Join your own breakout session."
I think Lorenzo wanted to join my group as well. I'm not sure if he can manually do it.
Sign in. You have to write your name because somebody have to allow him to reach.
Okay.
Marcela, Lorenzo Scipioni.
Okay, so we have the sections now. Hi, Susana.
Hola, Susana.
Hola.
You are mute.
That's good. Keep her muted. Keep her muted.
I can hear you actually, because you are in the room. I don't see appear in the breakout section.
So I don't think. Yeah. Yeah, we don't have customers.
I can just join Dave.
Yeah, I can stay with Human or with somebody else.
Yeah.
Okay.
Okay, you have somebody.
Yeah.
I do. Enrico, but you can join if you want, you know.
Yes, I know. So maybe we don't have anyone because nobody wants to join.
Well, you could unmute Lionel, and anybody wants to ask anybody here.
Yeah, absolutely. We can unmute to.
I'm unmuted and I am present, although my video is no. Okay, now the video.
Yeah.
Boy, look at the gang on top. It's the LFD, former LFD.
I like your shirt, Dave.
Thank you. Sammy will be appreciative because this is the second of the 3 Aloha shirts she makes me wear for these courses. So
Manuel,
tomorrow I don't talk and I may wear an LFD shirt.
Manuel, I think you have a question for Suman.
it's a comment because I can hear all the presentation, but all the images, it was very interesting. Sorry, my English is not very well.
Don't worry.
But I don't know what you think, Suman, but it's very interesting, the link that you do between the aerobic metabolism and the redox state. I don't know if you had a little idea about the study that I think that is from 2060. It's a few years ago about the this Dutchman called the Iceman that has all this technique routine, and the measure show that the ability to support the extreme cold is correlated to upper rate of consume of oxygen. I think that is a great field to measure all this stuff that you measure in that condition. That is my comment.
Yeah, I mean, that's actually great. Most commonly, I don't think I have seen much of a hypothermic experiments because most of the microscopes are not built to do that. If you look at any of the heat things that can change the heating conditions, right, in a microscope, they are always going up to 37 or 40 from the room temperature. There is very little which lets you come down especially in a commercial microscope setup to hypothermic conditions. And I think that's probably one of the reasons that hasn't been done. Means Indico and Davis, they were here, so they would know more about other studies, but I haven't seen any. I think that's the reason.
If you did this in Illinois in the winter in the old days, you just opened the window. Then you've got the very low temperature. You're not going to do this in Hawaii, I'm telling you, but
well, it will cost you more. It will cost you more because you need to refrigerate.
Well, we need to go up to the top of Mauna Kea, so that's another story in the winter.
In regards of comment, in here in the Institute Pasteur, there is a group working on metabolism and some drugs, new drugs that they discovered that they can change the metabolic pathway. Well, they do not change the metabolic pathway. They basically activate some metabolic loops that produce heat. And this is basically a new way that they found in order to, for example, reduce all the associated disease with the obesity, with diabetes, with all the things. When you were talking about this increasing in the heat, this remind me this because it's in the other way around. Instead of heating the person or cooling the person, basically you can activate the metabolism. And the drive force for them to test this is to put the mice in the cold room. They put the rat or the mice in the cold room, and this increase basically the metabolism in order to the cold. So yes, I think you are.
The reverse. Lionel, the reverse is true. Like in so there are these chambers which keeps the temperature at 37 degrees. So these are from mice, right? And if you put the mice there, they gain weight. I don't do any of those experiments. I've seen the results. They gain weight, and if you put them in fat diet, they gain more weight. The thing that I never mentioned in these talks is this is like a model of non-alcoholic fatty liver disease. That's why people use all this Western diet model. So, it's liver injury without having any alcohol in your system.
You not tell the public, but this is what Mariana suggests to me during winter, to use less dress to try to reduce the weight.
How did that work, huh?
You know, it's not related. Go ahead.
One thing I want to say about the question to Suman is studying metabolism. So metabolism is a general disease or is related to almost a large number of diseases. And really, at least in the United States, we are funded for a specific disease. It's very difficult to get funded for something general, and that prevents very much the use of or to do research based on something which is not specific. So anyway, that's it.
Yeah, that's why. No, that's why all this work is on the diabetes models and
Yeah,
this
because
non-alcoholic fatty liver disease model and things like that.
Yes.
And also,
Yeah, we can all.
about your model of the hi-fi diet. We are running now a project about hi-fi diet mice, but well, we also have the controls, so regular or wild type mice that we provide this hi-fi diet for 12 weeks. But also we do have one knockout for the ApoE lipoprotein.
Yeah, I know, I have them too.
And they have really a strong phenotype because they become atherosclerotics, and they do also have a really problem with the fat in the liver and in some other organ. And you know that I am particularly interested in the lung because I have been working with the lung for a long time. Well, we don't yet have analyzed anything related to metabolism, but we are producing a stimulus to basically make an effect or make a distortion of the lung, which is instillation of lipopolysaccharide. So the mortality of these mice are more than 50, 60, 70% of the control, just because they have absolutely dysregulated all the lipid deposition in the lung, and they are completely distorted. Yeah, it's unbelievable. So I am pretty sure that the metabolism will tell you quite a lot.
It does. I have seen some. I have ApoE mice before. I have used them. I haven't analyzed, there's none in the paper or anything. But if you look at kidney, there's a lot more fibrosis. If you look at metabolic changes, there are a lot of changes. So it actually is more evident. So there's a big study. Here it's more. I'm in a biochemistry department, right? So there's more of a medicine department, part of medical schools. So there's a lot of people who work on different models of fatty diseases. So there's so many different models that people have data for. The other thing that is kind of cool now that I started seeing is, and in terms of metabolism is, so you take the liver, which is a little difficult to do when they're frozen, right? So we never really measure fixed liver. So now I started imaging these fixed livers, and I think Andy will find this interesting that you see the vein and the artery, and you see the areas next to the vein and next to the artery, and they have very different metabolism. So there's a whole metabolic gradient from the vein to artery. So finally have the data, so I have to analyze them now. But just by looking at it, it's very evident, which I thought was very interesting to see. Even in a fixed sample, we could see this. Basically how the metabolism is changing in different parts of the tissue, just by looking at that, nothing else. That's, yeah. It makes sense, but it's just things are not never published, right? How many things we thought make sense, and especially including phasers, and then turns out not to other people, to us maybe.
I have a comment, nothing to do with metabolism, but it has to do with temperature. And it actually, I think it was interesting. As some of you know, I've worked for some years with NASA about they're sending a probe, planning a probe to go to Europa, one of the moons of Jupiter, land on the surface and do Raman mass and fluorescence. And I said, well, great. And I actually went out there. We were sitting in a committee talking one day, and I said, let me ask you, what's the temperature the sample is going to be at? They said, does it matter? I said, well, yes, it can matter. But what will be the temperature? And they talked and they said, well, it'll just be the ambient temperature. I said, what do you mean, the ambient temperature on the surface of Europa? They said, yes. I said, what is that? They said, it's about minus 150 degrees centigrade. So they said, what's the difference? I said, well, you're talking about looking for biological molecules in fluoresce. At that point, you start getting tryptophan, and even DNA things will phosphoresce at that low temperature, you know? And it's like, oh, they never thought of that. And I said, well, it would be nice if you could look at the sample and then warm it up, and then you'd see something disappears. You're seeing phosphorescence. And anyway, I don't know. Everything sort of stopped on that until Congress approves money. But the idea of temperature minus 150 on Europa, so that's another story.
Yeah, I remember when you were in the sabbatical at the LFD, we were talking about this. Did you already include the sine and cosine filter to do a spectra there?
Yeah. Unfortunately, we didn't, although if we ever get back to another meeting, I would definitely do that. But what was really interesting, I went up, as you remember, while I was in.
Now it's easy, just 3 filters, 2 filters and 1 total.
I went up to Seattle to be on a committee reviewing NASA grants, and they were going to different planets and all over the place. And my gosh, it was the most interesting one. Again, nothing to do with it, was a probe to Venus. The Russians had said 26, but the longest they ever lasted was a minute in the temperature and pressure. So the big goal was to spend hundreds of millions of dollars and get a probe that would last 1 or 2 hours on Venus. And I thought, wow, that's a different concept of spending money than we have, even at the LFD. So at any rate, maybe we'll be doing all phasers on Europa someday.
With the temperature thing, the book is right behind you.
Yeah.
Sorry,
one thing you have
I was just saying that
is, and nobody asked, is about the sensitivity of the wavelength measurement. So in the phasor representation, you have a point. And if you use any other thing, you have a spectrum. And so now how fine the movement of that point can be measured. And in our experience, more recent experience, it's about a nanometer. So that means that some of the probes that we have, or you have been using, can be used to be sensitive to the environment, for example, pH and many other things. So if we learn how to use a nanometer, a shift, we will be able to use the same probe, but for many, many more parameters at the same time. And that is something that I don't know why nobody asked the question of how sensitive you are. And you show those sine cosine filter, which actually have zeros. And so zeros make those very long distributions. But nobody asked that. But in essence, it's a point. You can reduce everything to a point. And among that point, you can see very important difference. And there is another work that we have been doing, which is using a bioluminescence probe, because in bioluminescence probe, you cannot have a very large spectrum. You have a spectral sensitivity. So depending on the bioluminescence probe where it's placed, that can have like an entirely new way to do bioluminescence. Not just to measure a color or whatever, but just to measure a shift of a color.
Yeah, absolutely, Enrico. Indeed, what the point that you bring is interesting also regarding of the number of channels that you have to collect for the traditional spectral phaser, and we have discussed this for many times, that how many channels you need in order to get the proper transformation, and how much you change the resolution in the phaser regarding of how many channels we put. And in this way, in this sense, what you say is the same. I mean, if you have 32 or if you have, 64, it doesn't change really much.
Yeah. So in the new software to analyze the sine cosine filter, I can expand. I wrote a routine that you can take a region of I don't know, 5 nanometer, and you expand and make the entire circle. And of course, you get much more sensitivity there, because you see sites in a cell or in a tissue that are different by a nanometer.
Yeah.
So that is very you can multiplex much more.
Absolutely. Yeah.
So anyway,
it's interesting as you know, when Gregorio Weber was doing all his work on spectra, he was using often center of mass of the spectra.
Yeah, of course.
He pointed out was much more sensitive. Nobody ever seemed to pick up on that, but we thought, wow, that was amazing. He could get plus or minus less than half a nanometer using the center of mass. And he would love this spectrophaser nowadays because that's really what he was doing in a cuvette.
Yeah, absolutely. But this is what I want to say. It's opened up an entirely new field. And for example, for the bioluminescence probe that you can see through the tissue or to an entire mouse, that would be very important, because you can see the difference might be in an organ, more acidic or less acidic, or more whatever, rigid or less rigid. So they are all new things that you open. And that is about temperature too, because the temperature, of course, changes the lifetime and changes the spectrum. And so you can measure, you can do a microcalorimeter, but in the entire body. So anyway, this is a possibility.
Dave, just for the temperature thing, your book with the cover of the temperature sensitive dye is right behind you.
Or that.
I know.
That's quite a difference in color, but it's very nice.
I actually have in my freezer Loredan and glycerol, and on the desk Loredan at room temperature, so I can actually show people right there. And then I say, look, it shows it in the book too.
So you can cook with Loredan, yes, and to measure the temperature of the turkey.
Here's a stupid question I thought of while you were just talking, that we know scattering of light, of course, blue scatters more. And I was wondering, if you were looking at the emission of a probe deeper in a scattering tissue, would you get a spectral shift because you have more blue scattered? Could you see that on a phaser? You know what I mean?
Well, not in the diver, because we try to collect the light.
No, in the diver you avoid that, but I'm wondering how that would look in a regular microscope.
Well,
if you could actually the same color put into different things, and more scattering, you should shift the phaser, wouldn't you?
Well, it might be, yes, but I don't. So the point is, you can form an image or not. Without an image, it will be difficult to make any conclusion, isn't it?
I'm just giving you the idea, and I expect next year to see all the papers on this new topic.
Well, so those beads that are shown, those were in scattering media. They appear.
Did you do spectrophasers to show the difference in when you get more scattering? How you must shift.
We didn't, and so in fact, if you look in very much detail on the points that Nick has, the points are elongated, are not really one specific.
Right.
So maybe we could examine.
Maybe that's due to the scattering, huh? Of the spectra.
I say could be. I'm not so sure.
Well, I'm not so sure either, but that's for me to postulate ridiculous things and you to do the experiment. So
yes, maybe in Europe.
Yes.
Okay.
Right.
Okay.
Say hello to Herman too.
So,
Hi, Herman. He's muted.
It's time to go back to the meeting.
Hola.
Hola.
Well, everybody's muted.
Hola, Susana. Hola, Germán.
Hola, ¿cómo estás, Alex? Hola, Suman.
Hi.
Nick, hola.
Okay, so we will continue with the last part of today, the tree application talk. And the next speaker is Dr. Susana Sanchez from University of Concepcion, Chile. She's associate professor in the polymer department in the School of Chemical Science. And today, Susana is going to talk about the application of phasors to measure ion concentration. And in particular, I found very interesting this work that he have done long time ago, and I always ask her to share this with the student because it's a very, very nice application. So Susana, I pass the word to you.
Okay. Can you listen fine?
Perfect. And I am at your screen.
Yeah, that's fine. I will just move it there.
Go ahead.
Okay. Okay. In the title, it said that I was going to talk about pH and calcium, but in just 20 minutes, sorry, I cannot do it. So I will just do calcium. And but it's similar to the pH. So we'll do film phasor analysis of calcium concentration in skin and in red blood cells. So the calcium gradient in skin is a work done by Anna Celli, 2010. And let's start like we did yesterday with the biological question. So the question was, this is a diagram of the skin layers. And we have the stratum corneum and the granular. And what they want to know if there is a gradient of a calcium concentration in, let's say, a stratum corneum plus a little bit like 28, 30 micrometers from the surface inside the skin. And if we can detect this with lifetime imaging. Leo, should I admit this person? Oh, would admit. For some reason, I can, okay.
You don't have to admit them, Susanna. That'll happen automatically, I discovered.
Okay, good. Thank you, Dave. So what we decided to use for this was calcium green 5N. And why, and this is for the student, why we decide that when you choose a dye, you have to see which one is the concentration you are going to measure in your tissue. And in the smooth muscle, concentration of calcium is around 1 to 10 micromolar. So the calcium green KD is around 14 micromolar. So that's the one we decide. So normally, this dye is used to do intensity measurements because the intensity go up with the calcium concentration. Now, the problem with that, with the intensity measurement, is that you can have this inhomogeneous labeling. And then in that areas where there is more concentration of the dye, when you take the image and you have more intensity, you may think and you may misinterpret that you have more calcium. With film, we don't have the problem. Lifetime is independent on the concentration of the dye. So then that's one advantage for the lifetime. And in the intensity measurement, what they do a ratiometric measurements to avoid the problem. And they can do it, but we have film. So we do film and we do 2-photon excitation, exciting at 820. And we will do phasor film analysis. Now, the first thing we have to do before seeing the data is to see how we're going to look at the data. So when you want to measure ion concentration in a tissue, you need to have a relationship, a calibration between the lifetime you're going to measure and the calcium concentration. So normally what you do a calibration curve. You take different concentration of calcium, put your dye, and then you take, in this case, your lifetime. And you get a binding curve, and that's like this one that we have here, where we have, in this case, the lifetime of the free species and the lifetime of the bound, where all the calcium is bound. Now, characteristic of this curve is the span, the difference between the lifetime of the bound and lifetime of the free, and then the KD. And those are the two things that you have to determine to do the calibration. How this binding could look in the phasor space. It will look like this. So it looked like a line where we have the lifetime of the free and the lifetime of the bound, shorter and longer. These both are non-mono-exponential, and the span will define the distance between them. And all the points in between, I mean, the one that we have here, will be in between these two phasors, meaning they're all the linear combination of the free and the bound. Now, what about the KD? The KD normally is a big problem. What people do, you try to mimic the media where you dye is going to be inside the cell. So you put BSA, you put buffer, you put culture media, you sometimes go to the extreme of destroying the cells and putting some cell juice there to make the calibration. We didn't go to that extreme, but we put these three situations. And what are the difference in the binding curve? The span. The KD is pretty similar to the one that it was reported. But look at the span, they're all different. How do they look in our phaser? Like this. So we change the slope and also the, let's call it size, the difference between the free and the bound. So we need to find a good calibration code for our system, a good span for our calibration system. And of course the KD too. Now what happened with the KD? This is a simulation that we move the KD, we increase the KD, and how does it look in the phasor plot? In the phasor plot, it doesn't change the angle or the span of our calibration, which is good. So no matter which KD we found, providing that is properly, I mean, sort of correct, we don't need to change much. We just need to determine the KD and found the proper span. Now, going back to the experiment of Anna, he took these hairless rats, they are newborn. You put it in a water bath, 37 degrees, and you put the dye on top of the skin. Then the skin is sacrificed. You take the skin and then you put it, you make a sandwich between a oversleep and a slide. And then we're going to do 3D imaging in the condition that I mentioned before. So this is the first image of it's pretty much in the surface of the skin. And this is the phasor plot that we obtain. A blob that is pretty much linear. We can see a free species and the bound species. So this is a pretty nice finding because we have an internal calibration curve. That's what we have. We have our own span here. What happens if we go deep in the tissue? We found that the slope of at each depth was equal to the slope of all of them combined. So we can use the same calibration curve, internal calibration curve, for all our analysis. Now we do have the KD, but sorry, we do have the span, but now we need the KD. How do we calculate it? Same thing. How do we calculate in the normal way? We realize that it doesn't matter how much we change the solution, the KD is going to be pretty similar. So we just found different, we just did it in buffer. We bought different concentration of, solution with different concentration of calcium. You can also make them, but it's a little bit tough to do it. For chemists will be fine. And then you take the lifetime of all of them, and then you found, like always, the fraction bound, and then you tell me your KD. So our KD was 12.2 micromolar. And then in the program, the only thing that you need to add is the span, that we have the internal one, and the KD. And in that condition, then we can go and analyze our image. And here we use what the people mentioned before, the reciprocity principle, and we have, and this is micrometer inside the skin, and this is already calibrating the concentration of calcium. And we do have that in the surface, there is like 0.1, 0.6, even 1.8. But if we get deep into the tissue, we have higher concentration. So this is just a resume of the data. There are more data, right? But the conclusion is this one, that there is a gradient of calcium concentration from the surface to 28 micrometer from 0.1 to 10 micromolar. Let's go now to single cells. So this is a work that was done by Vanessa Erlach, she's from Argentina, in red blood cells. One of the this is the biological question, like always. One of the strategies of E. coli to infect the urinary tract is the release of a toxin called hemolysin. And of course, the erythrocyte will be the first cell to be target of this toxin. So she was studying them. What was known at that time? Most of the process of the steps of the hemolytic mechanism of the toxins are well studied, not well known, but a lot of people study them. Now, what happened in the sublytical effect, meaning before the erythrocyte get broken by the toxin, there is any changes in calcium there. Why they want to know now that? Because that can be related with the mechanism of action, essentially. So our mission was to determine changes of calcium concentration before hemolysis. For that, we use rabbit erythrocyte. We just bought it from a company. And we use calcium green 1. Why we use that one? Again, the KB is 0.19, and the erythrocyte concentration of calcium is around 0.1 micromolar. We use 2 photons at night 20. We use FLIM, and we are going to do phasor analysis. Now, the first thing is like the example that I showed yesterday. You have to normalize this. We are going to work for probably a month taking data. We will have different supplies of blood, so we have to normalize to be able to compare the data. So our normalization was first, we standardized the blood as soon as it arrived, and we just fixed the hematocrit. I don't know exactly. And then we have to do an hemolysis, meaning we have to fix which one will be the concentration that will be sublytical for that particular blood that we get that particular day. So you just do a simple experiment, same amount of blood and increasing concentration of hemolysin. Make a curve, and then we choose before the hemolysis starts. And that was standard for all the samples that will arrive. We determine our KD in solution. We get the calcium solutions, we bought it. For the students, the calcium solution, you don't use too much and you can actually fix them. So even if they're a little bit expensive, they're really nice. So our KD determined in this way was 0.76 micromolar. What did we see? So here is the time 0, meaning there is no hemolysin here. And the first things that we saw is that already in the erythrocyte, there is an heterogeneous concentration of calcium that goes from 40, so here I just put, again, 2 cursors with different colors. This one will be from 40 nanomolar to 8 nanomolar, and the green 185 nanomolar to 40 nanomolar, the opposite way. So the calcium concentration originally in the samples are in this range, 8 to 185 nanomolar, with an average of 33 nanomolar. What happened when we put the toxin? If you look just the images on top, you will see that the color will change from more reddish to the red disappear. The red disappear, The green also disappear, and then it appears some bluish. In terms of concentration, what does it mean? In 78 seconds, we have that the concentration of calcium increased from 8 nanomolar in this range, 8 to 40 nanomolar, to 188 nanomolar, close to 400 nanomolar. So we could detect an increase in the calcium concentration inside the cells before the hemolysis. When did we stop the measurement? When they start to explode. I mean, no matter what, at the end, it is sublipidal, but too much contact, then will get destroyed anyway. You can also quantify, you can get the histogram of the images and then take the center and then have some nice curve to do some quantification if you want to. And you can also go and follow the kinetic of each independent erythrocyte. And the biological, the conclusion about this biological problem was that not treated cells, red blood cells, present a distribution of calcium concentration between 8 and 185 nanomolar, with an average of 33 nanomolar, and the addition of 0.54 micromolar hemolysis will produce an increase in intracellular calcium from 40 to 200 nanomolar in 78 seconds before the hemolytic process. And like yesterday, I just would like to make some conclusion about the technique at that time when we applied the first time for this system in the film phaser and the analysis for ion concentration. Well, the inhomogeneous labeling problem is solved not by the phaser, but for the lifetime property. We can obtain a detailed pixel analysis with the phaser analysis. Before, you used to have just an image, and you could do average analysis, color analysis. With the phaser plot, we can just go in any details we want to. And of course, the calibration, the span, we do have we did found an internal calibration, which is a pretty nice thing with this system. You don't have to start doing further experiment to try to fix the span or do a correct calibration. And the key value, you can take it from the solution and you will be pretty fine with the data. And I think that's it, Leo. Okay, finish.
Great, Susana. Thank you very much for the talk.
For 20 minutes.
You are great. You are the best. Make my life very, very simple and easy.
I know.
Susana, well, we will have time for question. If you wanna ask a question to Susana, this is the opportunity, because later on she have to go, and I think we are really good in time. So maybe there are questions in the audience. Well, they already have one.
Right. So Susanna, I remember in the old days, people doing red blood cells. There was always a problem with the absorption of hemoglobin and scattering. Is there an advantage with the 2-photon laser that you don't have so much difficulty doing spectroscopy on the blood cells?
Yeah, you do have, Dave, you do have to do what? This is for all the systems. You do have to study your sample without the label, and you have to find the wavelength where you can see your dye and then not see the autofluorescence. And in fact, with that experiment in the red blood cell, we bought erythrocyte from rabbit, from sheep, and from horse. And we did all the experiments, and the autofluorescence of the horse was so big that we couldn't work at any wavelength. We don't know why. According to Enrico, they were eating some grass.
Maybe you had a horse of a different color, like the Wizard of Oz, you know.
It can be. But so you really have to do that before. So that's a good question for the student. Yeah.
Okay. So.
Okay. There is a question here in the audience that say, what microscope did you use for the 3D imaging?
The normal, the 710. No, we didn't have the 710. No, actually not. We didn't have the 710. We have a homemade microscope, one of the old microscope. It was M3 for the reference.
M5.
Yeah.
The
But
M3
you can do, but now you can do nice 3D imaging with any of the microscope, the 710 or the 780, right? Yeah.
Yeah, the M3 was based on an Olympus, I remember so far.
Yeah.
It was the only one to have.
The size body.
It was a size body.
It was the size body. It is now behind the 80, right? It was the one with the Becker and Hickel and the flip box. Yeah, I remember. Yeah. There is another question from the audience that say, was curious, can this method also be used to see the local distribution of protein oligomers on the cell surface, e.g. Local distribution of monomer, dimers, or so on the surface?
Well, you could to see two things that are different, you have to have different properties on both. If the lifetime, in this case, if the lifetime of your monomer is different from the lifetime of your dimer, yes, you can do it. But you have to find the condition of what you want to. Yeah, you have to have two things different to see something.
But your hemolyzing was not. You didn't have any dye. It's just a.
No, we put calcium green, Enrico. Calcium green 1. We want to see calcium inside.
Because it's green, yes.
Yeah, no, we didn't want to see an interaction of hemolyzing with anything. Hemolyzing was just to produce the damage.
Yeah.
We were just looking at the calcium.
Mm-hmm.
And Susana, regarding the timing, because usually for the calcium signal, you know, there are different kind of current that you see. So usually lifetime takes some time because you need to accumulate some frames to get the statistics. So, so how would we basically to compete with basically the intensity, which is just 1 point, you know?
No, I don't. But at least with the instrument we have that time, we couldn't compete with those spikes. No, we were looking at the long-term calcium. Yeah.
You know,
we try to we optimize the power and the concentration of the dye to try to take less average each time, but you need to take average. I mean, there is no way to get a good signal. Yeah, no, but that is a disadvantage.
And do you know if there are new biosensors or just fluorophores that can change the spectra for the calcium?
For the calcium, I don't know.
Yeah.
I don't know. Is there any, Enrico?
Most of the time in the
but Roger Sien synthesized. It's a biosensor made of
the biosensor.
Blue cyan and the yellow fluorescent protein.
But FRET is a FRET one with the calmodulin.
It's a calmodulin, it's a FRET test. It's a biosensor.
Yeah.
But the range is about 15%. The range of the dyes that Susana was using is very, very large. So the lifetime changes a lot.
Yeah, but Leonel was asking if there is anyone that will change the spectra depending on the calcium concentration.
From the blue to the yellow.
Yeah.
Okay.
It's a isometric probe.
Well, there's lots of those probes I mentioned in my first talk that have big shifts in either excitation or emission. I mean, I forget which ones, but the Furas, the Fluors.
Oh, that's true, Leo. The normal one. People
have a lot of those probes.
Yeah, the ones they use people for ratiometric, like the Fura, I think.
Yeah, but
they all have.
You cannot use in the microscopy.
Yeah. In this case, we were looking the one that just increases in intensity but doesn't move in wavelength.
Right.
Okay, great, Susana. I think we don't have more question. So thank you very much.
Well, thank you.
We will continue with the talks, which are my
nice short day.
Thank you. Sandy will be happy.
Okay, let me share my screen. I will give, basically, we switch with Suman because I thought he will make a great presentation as he did. And I will just focus in a particular development that we did when I joined LFD, which was related to the measurement of absolute concentration of NADH. But this is also true not only for the NADH, it's also true for any kind of a fluorophore that we have in the cell and that we can use as a fluorescent biomarker. We need just to do the proper calibration. So the question that I place here is, well, how we can get NADH absolute concentration in vivo by FLIM? Well, in this is a contradiction, because from the introduction of then when Enrico presented the phasors, and he again emphasized about the independence of the lifetime to the concentration, to the change in the concentration. So how this can work? Well, you know, I will try to address this, basically explaining how we use a light to interfere the fluorescent lifetime or the amount of fluorescent lifetime coming from the NADH with a third component. And I will explain in a second which was this third component and why we pick this. So as I said before, why we would like to do lifetime? Well, it's a very characteristic property of each fluorophore. Many factors can affect the fluorescent lifetime, like ions, pH, hydrophobic properties, oxygen concentration, because it's a good quencher, as Suman addressed before. It also changed by binding, like the case of the NADH. Upon the binding with the enzyme will change the lifetime. And also it changed when you have molecular interactions, such as FRES. So, well, lifetime is a very useful value or technique that we can use for generating contrast and basically do imaging with this. And I place here a couple of examples, one related with the solvatochromic probe Nile red, and these are done with lifetime, and you can see basically all the organization of the ER and also the mitochondria, the nuclear envelope, and the change in intensity means change in the lifetime. And this is a biosensor, it's RAC1 biosensor. And this is done in collaboration with some Amy Edinger in UCI, and we can see the formation of macropinocytosis of these cells along the time with different stimulus. And this is a very nice application that we can find using FLIM. But one of the main thing that for why we always claim for lifetime instead of intensity measurement is because it's independent, and we don't need to think basically that the distribution of the fluorophore that we are studying is homogeneous in the cell. Well, I place a question: is always independent of the concentration? Well, I will tell you more with this example. Well, NADH is important because it's basically is heterogeneous distributed across the lifespan. So all live cells has NADH. And the good thing is, well, we don't need to dope our sample with any fluorophore. Basically, it is already came with any cell. And basically, it can be split between two populations, the free and bound NADH, depending on the interaction or not with the enzyme. This change in the interaction between free and bound, it changed the lifetime. And these are part of the experiment that Suman showed today. And this was very well described and correlated with the optical redox ratio versus the seahorse analysis for the consumption of oxygen. You might notice that even when lifetime is independent of the concentration, when you have two fluorophore, it's true that you have basically amount of each of both, and this will be affected at the final situation that we have in our sample. Because if you have more than one, you will pool the amount of the fraction that you are collecting regardless of the lifetime. The lifetime is still the same, it will not change, but you will have different fraction. So can we stress this in order to use this method to calculate the absolute concentration. And why absolute concentration of NADH is important? Well, basically, as we already know, this trajectory between free and bound NADH we call metabolic trajectory, and the change toward to free means that we have higher glycolytic condition in our cells. When they move back to the bound NADH, it means that the cells are more close to oxidative phosphorylation, but the concentration is not present here. So how can we measure this absolute concentration? Well, I will try to convince now about how we address this question. So Enrico one day come to me and say, well, Lionel, if we can interfere this system with a third component, and this component need to be something that we can add to the sample, and it's better if we don't need to add something which is that we need to poison. Maybe we can do physically, but in a different way. And he said, well, there is a point in the phasor which is very simple to do. And this point is the one that I am pointing here, 0, 0. 0, 0 means infinite lifetime, so unmodulated light. So he said, well, if you open the room when you are doing the experiment, this will give you a chance. And maybe we can basically interfere our system and quantify after the calibration how much we have of free and bound NADH in our system. So how I started? Well, this was the first lesson that I learned from the LFD. This was really after a few months. And I jumped to the left trying to be, learn something of building stuff. And I'm very proud of this. I made this gadget, which is basically a flash lamp that I found around. And then I place a diffuser, and then I place a bandpass filter to select different colors. I was trying to use this also for the Laurdan. Then I place an iris so I can measure, I can change the intensity with different step and different size of this iris. And I place ND filters also to reduce the amount of the light. So I place this on top of the microscope, and then I start to do measurement, and nothing works. And I came back to the Enrico's office, and I say, well, Enrico, this is not working, this is not linear, this is not going to work. And they say, well, can you explain me what you did? So I say, well, I put this, and this, and I say, can you tell me what is wrong? I was trying to explain that I pay a lot of effort to place this diffuser to make basically the most homogeneous that I can, the light and the iris, and say, well, Lionel, this is not going to work. And it's not going to work because either changing the 80 or either changing the power of the light, you will change the spectra of the response. And I say, if you want to do it and you have to do it properly, you have to use polarization. So basically, we built just a device that we place in the regular transilluminator. We place 2 polarizers, and we modulate the light just using the polarizers, as most of the old microscope works. And indeed, is the way that I am using today for the building of my 2-photon microscope. I'm using polarizers. And this enable us to continue with the project. So what is the idea here? Okay, imagine that we have a solution of free NADH, that we can dissolve NADH in a buffer, and then we can do a measurement. And the measurement will be in this position, which is more or less 0.4 nanosecond. If we shine different amount of light, the position of the phasor will be moved toward 0, 0. Because as much intensity of the light, we will compete basically with the fraction of NADH and our light. So if we do that and we calibrate our system, so how much is this step, how much is this step, how much is this step, and then we place everything in our software, we can determine where is the shift and how we can calculate the absolute concentration of our system. Okay. Well, I was very happy because everything was working, and Enrico come back the next day and say, well, Lionel, I found another way. You don't need to use your polarizer. And I say, no, come on, Enrico, I spent a lot of time. I think this is a very nice construction. I am very proud of this. Say, well, look at this, Lionel. Well, if you think about, we can add electronically as much light as you want, because it's an infinite component. We can place in our system a third component with an infinite lifetime. And we did it, and we did the calibration from the measurement and the electronical light, and basically everything came in a line, which is very nice, because now we don't even need to do this calibration with different amount of the light and the concentration. We just need to take one record of the absolute concentration of NADH before or after we measure our cells. Okay, how this work? Well, we have our sample, we place our lamp, and this moves toward a certain distance, and we label this distance with the concentration that we know, and then we do the same electronical, basically the software does this for the bound. And we need to take care of something, because the quantum yield of the bound NADH and the free NADH is different. It's more or less 7 times different. And you may notice that the distance for the same amount of light that moves the position of free NADH towards 0, 0 is that long. However, for the bound is this little long. And this is due to the quantum yield. It's more hard to move the bound NADH toward to 0, 0 than the free NADH. But we can calibrate this, and the software does all the work. So then we have a measurement. This measurement were done by Nick Ma, a PhD student of Michel Digmar lab. And basically here we have the classical free and bound trajectory. And if we place a cursor, we can see that the short lifetime is inside the nucleus, which is something that we already know because there are no metabolic enzyme there. And then we have the long lifetime in the rest of the cytoplasm. And this is something that more or less we, at that point when we were doing the experiment, was known. And then we apply our lamp. And as soon as we apply our lamp, we notice that the concentration were different along the cell and more complicated than the situation that we have here. So first of all, we can see that there is a spread in the data, very long, and then there are some very bright spots here. So this very bright spot here, which are this red selection here, is basically a concentration of NADH, which is very homogeneous in all this region because it's very concentrated here. However, in the line, we have really a variance of concentration of NADH, regardless of the free and bound. We can also tell which is the fraction of the free and bound. So we can do first the fraction of free and bound, and then the absolute concentration of the fraction of free and bound. So you may notice that the most strong area, which is this one in red, are associated more or less where we expect to have the mitochondria, where the bound NADH will be more important. And the less one concentrated is here in the blue cursor, which is in the perimeter. And we know that there are some enzymes we use that use NADH and also NADPH because we cannot discriminate between NADH and NADPH in this experiment. So then we did this, we analyze, we make a scale to color the range for the concentration. And basically, after all, the story ends basically measuring the concentration of NADH in the whole cell with the pixel resolution on an in vivo cell. We did all the calibration and we were very, very happy because the concentration that we got is more or less what was described by biochemistry experiment. And this is very interesting because if you think about to do any kind of measurement of absolute concentration, traditional biochemistry need to distort the sample. You need to break the sample and do experiment in an analytical fashion. In that case, we can calculate the absolute concentration of fluorophore without even affecting our system. It's just by a calibration that we did before or after our experiment. And the only considering that we need to have is that we need to keep our system in the same condition, meaning same laser power, same calibration for the detector, etc. So this is conceptually what I would like to tell in this presentation. I want to thank, of course, Enrico for giving me the chance to work in this, and also to Michelle Digman, because this was a project of Ning Ma, a PhD student of Michelle, as I said before, and I enjoy a lot working together in this project. And we have the paper, and there are some other people working on this using the absolute concentration. I found very nice, interesting to add to the traditional free and bound NADH also the absolute concentration analysis. Here is Michelle and Ning and all the LFD troop. So I will stop sharing now, and I will give also the chance to the audience to have questions. I think we are very good on.
I have a discussion, a thing to discuss. So if bound to a protein is what changes the lifetime, how come you get a distribution of concentration? So the concentration, so if you have a given concentration of an enzyme and the given concentration of an ADH, the KD will determine how much is bound. So
Yeah.
how come you get that very large distribution? So there have to be some principle for that.
Well, yes, absolutely, yes, Enrico. Well, you remember we have discussed this quite a lot, and
yeah, but maybe something that interests everybody. So how come?
Well, the idea is, well, we have different amount and different location of NADH concentrated in specific domains or in specific places in the cell.
Well, you have to be sequestered. Essentially, you need to have regions which contain a different amount of enzyme and different amount of NADH, because otherwise the equilibrium will always be the same.
Exactly. Exactly, and this is also very important because you remember all the work that we, and all the carefully work that we did in order to have free and proper bound NADH, because...
Well, but we only use a free NADH, which is easier to obtain.
Yeah, yeah.
But the question is that a corollary, so a conclusion on those kind of measurements is that the cell interior cannot be homogeneous.
Yeah, absolutely.
It's not, but also you can tell where is different or not. And after you did the work, you know, we studied very carefully the mitochondria and the nucleus and the nucleus in different conditions and different things, and the cell at division, when the cell divide, what's happened, and so on. So it's really a method to look at the distribution of the concentration, which is not available with the lifetime. All the points are on the same line, but at the moment you perturb the system, you can start to see immediately that this is not true. Some points move and some points don't move.
Yeah, this is interesting when you think about the change in the free and bound. It wasn't that long, right? It was really concentrated in an area, but when you spread with the concentration, this get really long in certain areas of the cell, right? And this is what we are pointing out, right? So these areas, the only way that you can spread that long is because you have region where you can have quite a lot of bound and poor of NADH, free NADH, and the other way around, right?
Can I ask a question, Leonel?
Sure.
Just to anticipate what some students might be thinking, obviously there's more than one dehydrogenase in the cell.
Yeah.
And certainly mitochondrial malate dehydrogenase has a much longer lifetime. How much of this apparent change in concentration might or might not be due to different populations of dehydrogenases? Can you address this in any way? I don't know how to address it. So.
Well, you are right, Dave. Indeed, you remember when you were in the sabbatical, I take advantage of you because it was not that easy for me. I never worked before with enzymes, so and the preparation and the equilibrium that you need to have to pull basically the enzymatic equilibrium is very important. And of course, if you have, I don't know, I am thinking now in your famous mitochondrial malonate dehydrogenase, which depending on the condition that you prepare the sample. So if you add malonate, it will change the lifetime really much more.
The same as an LDH. When you add oxalate, it changes the lifetime.
Exactly.
So,
but the point there is is follow. There was some papers some time ago that they try to trace basically linear combination of the different free and different bound enzyme, and then try to estimate how much of each enzyme you have. Well, this is possible, but you need to think about that in a pixel you have more than one, right? So if you have more than one, this is not that easy to solve, because as Alex showed before, decomposing the fraction of multiple components, because you can have not only LDH and MDH, you can have many more, will be very difficult. So, but still, if you had, I don't know, 3 or 4, the pooling of the interfering light will make the work and will split by the quantum yield of each of them in the different component, right? So
well, have you ever been able to go to the detail? I think Enrico is doing some of this now to separate. I remember some of the people with the LFD, the mitochondria things from the cytoplasm, where you would have maybe a different populations of NADHs. Have you done this absolute concentration at that level?
Never did,
but there is no difference.
Really?
Yeah,
that's interesting, because boy, so somehow the succinate dehydrogenase and the mitochondrial malate dehydrogenase come out giving the same average lifetime or something. But I don't know. I think there's still some more to explore, but it's not an easy topic.
Yeah, but this is not what the data show. You know, in some way, the data will show that if we see a change in the angle of as we add the zero component, it will change the angle, but doesn't change. It change everything. Actually, the nucleus is very different than the rest of the cell, because the concentration, so the metabolism is very different. So in the nucleus, an IDH is used for SIRPIN1 and other, it's a substrate, but it participates to another sort of equilibrium. But in the nucleus, in fact, it's very homogeneous, but the cytoplasm is not. It's not, but
something to look at.
Difference that you have reported is maybe someday we will investigate that with more in detail. But in some way, you didn't study entire cells.
Yeah. Suman, I think maybe you want to make a point.
Yeah, I just wanted to say also, if you think about it, it's a tangential scale, right? So the changes between 0.4 and 1 nanosecond is much more than 3.4 to 5 nanoseconds. It's a much smaller scale. So differentiating at that part of the phasor plot is much less sensitive in a sense than differentiating on the others. And that sometimes works in our advantage, because a small change on that side doesn't change the metabolic trajectory that much, which means we can still look at it without being very specific about. Otherwise, you need to specify exactly what it is binding to, which starts a different sort of conversation similar to what we are having right now. Just wanted to say that.
Yeah,
well, that's good, though. There's still more to understand, so that's terrific.
Yes, that's the right conclusion.
Yeah, absolutely. Let me pass to more questions. Yeah. There is a question that say, in case of visualization molecular marker in the cell exposed to ionization radiation, it will be similar procedure. Think about oxidative stress. Well, I guess so. Yes, of course, the procedure will be, there should not be changes. About the oxidative stress, I think you can remember the final talk of part of the talk of Suman, in which oxidative stress pull the metabolic signature when you have lipids, toward to this long-lactane species. And this was a marker that the lab found a long time ago, working with Rupsa, and also Suman, because no other people ever saw this, because they were cutting basically at the end of the decay, and only when we can collect the full decay, this show up in a way. So yeah, I think there is no difference in the procedure. And then the last question before I move to the next.
Yes, Felipe Montesino.
I wonder if the highly dynamic nature of mitochondria, which make it kind of difficult to image precisely, will affect the determination of lifetime using the phasor, then move a lot. Well, this is true, Felipe, because I mean, the cells are moving and the lifetime are moving, but a lifetime measurement, if you have a bright fluorophore, it will take not that long, but still you need, I don't know, 20 seconds or something like that, or 30 seconds.
For an image, but not for a pixel. In a pixel, you can measure in microseconds.
But what I think Felipe wants to stress is the fact that if you have a point and in the next measurement of the pixel, the things move up or down, of course you will assume to this a decay. The decay will be an average of whatever is passing through this volume, right? And you cannot do nothing. But basically,
well, but you can measure one mitochondria in a few microseconds. So that is true up to a given amount. Of course, if everything moves, you get an average. But you don't get an average, because otherwise we will not see the mitochondria. We will not be able to find them. And yes, it's very dynamic. But, absolutely. But what Dave says is something different. And so either in solution, you have something different than you have in the mitochondria, which is possibility, but that have to be demonstrated in some way.
Well, in solution, the long lifetime is when you have hydroxymalonate bound. I don't know in the cell how much hydroxymalonate is bound. You know, it's completely different probably than oxalate binding to LDH. So probably that's involved in the answer, but
yeah, that would be part of the answer, but still clarify a point.
Yeah.
Okay, so there is no more questions, so let's move to the last application talk, which is also me. I will have water. And let me share my screen. And this talk is going to be related to Lardon. And the combination of spectral and lifetime will help us in this case. So as I say today in the morning, Lardon is really a wonderful dye, and I will try, if I have time, to make a point of this, because I think this is very important, and there are some confusion in the community about the use of Lardon, and I think this is very important to tell. In this case, what I wanted to do is the understanding of the effect of hydrogen peroxide in the membranes. And for address that problem, we use basically Lardon, and we image our cell using lifetime microscope and a hyperspectral microscope at the same time. And we took one image after the other, in a very short time. So we can consider that the image was taken at the same time, in a way. Okay, the lateral organization of membrane is very complicated, because if you think about, we have between 100 to 1,000 of different lipids within the cell. Besides of that, we also have cholesterol, which make our system more complex, as I showed before in the morning. And also we have proteins in the membranes. We have 100 or 1,000 of proteins. And on top of that, we have changes in the polarity or the activity of water due to the molecular crowding. So really, to address the problem of measuring the dynamic of membrane in the cell is a really complex system. And the point that I want to make here is, well, the use of fluorescent proofs that can sense particularly characteristic of the membrane are very, very useful. So one example of this is Lardon, because it can measure the membrane heterogeneity due to the change in the spectra and lifetime, as we saw today in the morning, by the by the interaction with the particular place where Laurdan is at the membrane. So I will not go, I think in the next slide, okay, we go a little bit in detail again about Laurdan, but I will want to stress this thing. Okay, I'm not an expert in oxidative stress. I just know a little bit, but I want to point it out that basically hydrogen peroxide was considered really a bad thing for cells because can produce damage. But in the last 10 years or so, there is a concept that emerged in the biology, which is basically the difference between oxidative stress and eustress. And these two things are different because one means that is really something that can hurt the cell, but the other are very basically fundamentally for the signaling within the cell. And the cell spends a lot of energy, and it has a lot of system, in simologically speaking, to control the concentration of hydrogen peroxide. Another important thing is the distribution of hydrogen peroxide from outside to the cell to inside to the cell can change in 6,500 or up to 1,000 times between the two places. And it's very interesting that there are some places in the cell where you have a zero amount of hydrogen peroxide. So if you have one molecule, it will make the work. And this is very important. And for this region, you have really a bunch of different enzymes that can reduce with different KDs the hydrogen peroxide. In particular, there is a group of enzymes which they are called peroxyredoxin. They mentioned in the beginning lecture today in the morning, because there is an important group here, work led by Anna De Nicola, that work in all the reaction of this enzyme with different oxidant that can react really up to the limit of diffusion. There are two models about how this signaling works. One is the flush gate, which means that everything got oxidized, and then when the first enzyme that reacts, the next one can react, and the next one, and this is a path of oxidation. And the other one, which is nowadays more accepted, is the peroxyredoxin sensor, which means that these react really, really fast, and then they transfer basically the information to secondary enzyme to make the work. And we will see that this makes a lot of sense with the result that we have in the paper that we published, because there are two enzymes which are very important, protein kinase C and phospholipase D. So one thing that we knew before we start this was that hydrogen peroxide can change the fluidity of the plasma membrane in yeast. And this produces a reorganization in the supramolecular structure of the membrane, measured by polarization in cuvette using the diphenylhexatriene. So as I said before, we addressed the problem using Laurdan, in which we substitute, the substitution in this point was a lauryl group. And this was designed, as I said before, by Gregorio Weber and Faye Ferris. And I want to make a point here about the importance of this Laurdan, this lauryl group. Well, this lauryl group give to Laurdan the tension to be a particular region in the membrane in which it can sense a few molecules of water who has restricted mobility in terms of rotation. So if you think in a molecule of water rotating in a glass of water, so the bulk water, it will rotate in the picosecond time scale. However, these molecules are rotating or moving on the nanosecond time scale. And this is very important because this reorganization of the water molecule around the dipole is competing basically with the emission of the Laurdan. And this is what we can sense when we analyze the dipolar relaxation. So if we can measure the lifetime in two channels, one in the blue and one in the red, we can split these two parameters, which are basically the polarity and the dipolar relaxation. And the polarity is related to the average dielectric contrast of the apparent dielectric contrast that we have in the environment of Laurdan, and the dipolar relaxation is the physical reorganization of the water around the probe. So this was basically what I was trying to illustrate. And the point that I want to make is if you modify the Laurdan with any kind of different structure, this will move Laurdan up and down in the location in the membrane. And at the end, you will be sensing different things. And it's not possible to tell that you have similar proof of Laurdan basically because you have the same chromophore without taking into account that basically the location of Laurdan is due to basically the fluorophore, but also the Lauryl group. And this is important because otherwise you will be sensing different things. And as soon as you change also the chromophore, it will change the distribution of Laurdan depending on the liquid order, disorder, or phase membrane. And Laurdan, one of the unique things is that it can diffuse across all the membrane without have any particular affinity for each of these. So I already went there. We will use basically this thing of measuring film and hyperspectral, and I explained how this works. So we will use NIH fibroblast, and then we will place it, wait for 24 hours, and then we will stimulate using a concentration, a bolus of hydrogen peroxide in 2 concentration, and wait for an hour and do the measurement for our experiment. So how we will analyze the data? So we will have 3 variables. So we will have one set of data, which is film, and the other will be spectrum. So the film we split in 2 channels. So for the channel 1 that I call blue is the range in the more or less center in 440. We will collect our data, and then we will analyze using this 3-component analysis in order to see the change in the fraction of the liquid order and liquid disorder, and then if there are dipolar relaxation. We will not expect to have dipolar relaxation in the blue channel. We will have to see dipolar relaxation in the green channel because it's related to the environment in the water. So the relaxation of water across the probe. So for this, we will basically trace what is the fraction of dipolar relaxation regarding one dimension, and what is the fraction of fluidity regarding regardless of the relaxation or not. And this is how we analyze the data, and this was introduced by Suman today. So if we do this and we treat our sample with hydrogen peroxide, what we immediately see is analyzing the full image, we didn't notice much difference. We see that there is a decrease basically in the distribution. And this distribution from here is basically the histogram of all the pixels along this line, right? Then we calculate the center of mass of this distribution, and we compare the center of mass for each of the cells between the 2 groups. And this is what I am presenting here. So we did the same for the dipolar relaxation, and what surprised me immediately was that we do have a change in the dipolar relaxation in the blue channel, and this was unexpected. Let's keep this in mind for the discussion at the end. When we analyzed this, but now using mask, everything became more clear. So when we do mask, we only focus our eyes to this region. And in that case, you can see changes in the membrane, and the changes are moving toward to less fluidity. So the membrane are becoming more ordered. The same happened with the dipolar relaxation. So the dipolar relaxation basically is increased when we have more ordered membrane, which is in a way anti-intuitive. So then we analyzed the second channel. The second channel was the green, and this will be focused mainly in the move out of the universal cycle, because it's a reaction during the excited state. So it will pull the data out of the universal cycle. Something similar happened, but in that case, you can see there is another component here. And when we analyze the center of mass, we do see a difference, significant difference between these 2 points. And this difference is huge when you compare the dipolar relaxation. So it's really extreme change toward to higher relaxation when we have hydrogen peroxide. When we reanalyze the data using the same logic, and basically we segmented and we collect the region where are more related to plasma membrane, this also happened again, and we can see a shift back to less fluidity in the plasma membrane. And then the most important thing here is the dipolar relaxation, and the same happened that in the whole cell analysis. But now we have also a trend by the different concentration of hydrogen peroxide. So as a bottom line, fluidity decrease as dipolar relaxation increase by hydrogen peroxide in the green channel. So, but as I said before, we have also spectra, and we collect the spectra, and in the spectra, we basically have the 2 variables, the fluidity or the order disorder and the dipolar relaxation together, moving along this line. In that case, we have a strong background. And as I said before, this is not a problem. You need to include in the system and then trace back where should be the position of the pixel in our linear combination. When we do this and we analyze the plasma membrane, you can see that there is a shift toward to increase in liquid order, which is in coincidence with what we get for the lifetime when we introduce the hydrogen peroxide in the system. And this is also significant when we analyze the center of mass of the distribution along this line. So finally, what we did was trying to connect the dimension of the lifetime with the dimension of the spectral phasor in order to see if we can figure it out what was going on between this spectral shift, the dipolar relaxation in the blue and in the red channel. So in the analysis, what we did, we placed cursors here, and these cursors were fixed. This is the linear combination for liquid order, liquid disorder, regardless of the channel what we were analyzing. And this is control and hydrogen peroxide. And then we did another linear combination for the dipolar relaxation. When we analyzed, when we found this pixel here, then we tried to trace back where was those pixels in the lifetime channel 1 and in the spectral phasor. And you may notice that the shorter lifetime here and here are the shorter lifetime in the other channel, and also the redder pixels in the spectral phasor. And the other side is also true. The green here are more toward to the plasma membrane, you can see here, and also toward to the longer lifetime here, you can see here better, right? And then it's moved toward to the liquid order here. In the dipolar relaxation, you may see that the most outside is related to the interior of the cell, right? In the both cases. And then is here on the universal cycle, and here a little bit outside on the universal cycle. And the same is true for the other case. So at that point, I was absolutely lost. And I came back as usual to the Enrico's office and say, Enrico, I cannot figure it out why we have dipolar relaxation in the blue channel. And as usual, Enrico came up with a very nice idea, which was, well, he already did some experiment with Tiziana Parasasi in this paper very early in Cuvette, the 1993, in which if you have fast interconversion in the system, which means if you have a domain, basically the time that take the Laurent to go from one to another will basically give you a final result that if you get excited in one environment, it will emit in this environment. it will not change. You will have on average not change from one system to another. Remember that the diffusion of Lardan is very fast, more or less in 1 micrometer square per second, and this allows you to travel that distance like this. Okay, if this is true, what he pointed out is, well, what happened if you have structure which are like filaments in the membrane? And this happened, for example, in shell membranes. And they demonstrate in this situation that when you have shell membrane, you can have excitation during the shell, but then the emission in the other membrane. And if this happens, you will have this effect of basically exciting in the blue, but emitting in the relaxed environment. And at the end, you will have relaxation in the blue channel. I found basically this really nice explanation for the result, but now we have to explain this from the biology. And the explanation that I found is this thing. Well, hydrogen peroxide can activate protein kinase C, and protein kinase C is activated by, well, phospholipase D can be activated by protein kinase C. And the phospholipase D is in charge of chopping basically the head of the phospholipid, making a phosphatidic acid. And this changed quite a lot the methyl transition of the phospholipid, making completely solid at the temperature where we ran the experiment. So in that case, what we hypothesize is that if we have this situation, I did some more experiment. I never finished this experiment in order to corroborate this hypothesis, but we did experiment with a sensor for protein kinase D, and we were trying to stimulate and basically inactivate PLD or inactivate PSC in order to see if the stimulation of hydrogen peroxide can reproduce the experiment that we did for Lardan. Still not conclusive, but basically if we have lines that the Lardan can get excited here, but then moving out, this is the explanation that we found for this application. And the interesting thing here is, well, basically using the best of each of the both methods, we can conclude something which is very difficult to interpret and to give us a solution, which is very, very, very important. So this is finally the end of my talk, and I am more than happy to discuss and answer questions if there is anyone from the audience. There is any question? No, I can. There is nothing in the chat. Yeah, here there is a question. Question 1, NIH323 fibroblast, Amit say that a specific cell type for this experiment, or can it be done in any other cell? No, this was the cell that I usually use in the lab, and there is no other reason that just I was a lazy person. That's it. That's it.
That was ready.
Yeah. Oh, okay, that makes sense. Yeah.
Yeah. Yeah, I want to stress something that maybe I didn't explain properly today in the morning with the spectral phaser. One of the unique of the spectral phaser, as well as the lifetime phaser for Lardan, but in particular spectral phaser for Lardan and Agdan, is that in the general thinking for global demixing, you need to assign a maximum and a width, and then you can decompose the component. You cannot do this in this situation because we don't know where should be the location of the Lardan at the end of the end, because it depends on the system.
Well, the shape of the spectrum.
Exactly, the shapes of the spectrum will change, and we don't know how it's going to change. Indeed, if you look, for example, Lardan or Agdan in a yeast or in a superior eukaryotic cell or in a bacteria, it changed a lot. So if you assume that the maximum will be here and then is there, all the things will will get that wrong. So this is really, really important. To have a model-less analysis is really valuable for this kind of application.
Well, in some way, people using generalized polarization hide the idea. You just make one color and the other color, and that's it.
Yeah.
So you pre-assign the thing, and then of course, it works, but it's limited. You cannot see details.
Yeah, and the other point that also I want to stress about, or I want to discuss about Loudan is the unique opportunity that Loudan provide in order to work in vivo. Because most of the people complain about Loudan is equally distributed across the cell. And in my opinion, rather than be a bad thing, this is really a unique thing that Loudan has. There is no accumulation of Loudan in any particular organelle by itself. It's absolutely equal distributed across the cell. And this is very important because if you want to really address this in a proper way without biasing by the affinity of your proof for a system, this is important. And the other important thing is the location. If you
also no partition.
No partition. If you think, or when you work with I don't know, other version of Loudan like C Loudan, M Loudan, or newer version of Loudan, and you think that you can use all that we know, all the knowledge that we have, because Loudan is really well understood, because Enrico started with this in 1987, and since then there are more than 60 or 70 papers from the lab explaining all the details and how this works. If you think that you can extrapolate everything that we know from Loudan to any other version of Loudan, this is a wrong thing, in my opinion.
Well, the thing is that the lifetime in water is negligible. Does not fluoresce in water.
Yeah, this is true. But if you have different location of the Loudan in the membrane in terms of depth, this will change all the things related to relaxation, right? So Dave, yes.
Yeah, I just wanted, maybe you should comment to people that the Loudan really started to become useful in cells when you had the 2 photon excitation.
It's also important.
Because with 1 photon, it was so photobleachable. Now, however, I'm reminded of the new probe of IPMON, who's on the screeners right below you, that is similar to Loudan, but is more photostable, and that would be something to see.
Yeah, absolutely.
And another topic, I just want to say this for the young people in the audience for future careers to think about. You know, now you can do these measurements under high pressure, elevated pressure, you know, in cells, even under the microscope. And pressure, as you know, really can affect this fluidity of the membrane. And some people might say, why do we want pressure? We live at 1 atmosphere. It's true, we do, but most life on Earth lives at elevated pressure. And unfortunately, I'm too old to get into this now, but I always wanted to get organisms from the deep sea and then study them to see how the pressure adaptation works. But this would be another future, I think, for probes like Loudan and the microscope, and but in a capillary tube, as Rico's done it. Imagine doing high pressure of cells in the microscope with these sort of probes. Really, that's a whole new area that could be opened up. So maybe some of these listeners will do that one day.
Indeed, Anna Celli already did some of this stuff of high pressure and QVs.
Right, but now we could do, it would be nice to do with cells, actually, so.
You also bring a nice point to discuss. Some people complain about the poor photophysics of Loudan because it's bleaching when you work with 405. Well, in my opinion, you cannot tell that the Loudan photophysics is poor when you are using 405. 405 is not ideal equivalent to excite Loudan. So you are really exciting at the edge of the excitation spectra. And we all know that Loudan has this red edge effect. So if you change the excitation wavelength, you will you will excite different ground state because Laurdan has different ground state. This is another big part of the contribution of Tiziana and Enrico doing excitation spectra instead of emission spectra.
The two photons are getting so common that I think people will do that. And again, maybe we'll learn some more about Herman's new probe as time goes on too. And
yeah, I think Thursday Herman will speak about this new probe that we were comparing. We have a paper very nice about Caprida, and it is a very nice proof. It can also sense dipolar relaxation, which is really interesting because
it'd be nice. Remember I showed the PubMed citations for DPH and shifted in phase was Lordan. Maybe Caprida will be going up, displacing both of them someday. I don't know, but
yeah, because it's visible. You can excite it visible. That is an advantage.
The good thing, you can excite 488. I mean, any confocal microscope will have 488. Yeah, exactly. And we did a very nice comparison, in my opinion, between Laurdan and Caprida, and we found really a very good, interesting probe.
Well, we'll let Herman talk about that. Is that tomorrow?
It's Thursday.
Thursday, okay. Looking forward to it.
Yeah, Herman, you are there. You will have the chance to present all the data. I don't want to.
Okay, so today we finish now, or we have a breakout?
No, we finish now because Susana have to leave. If there is no more question for any of us, not only for me, if there is question for Enrico, for Alex, for Suman, Dave, I don't know if Nick is still there, but we are all welcome to answer any question in the audience. If not, we finish for today. I really thanks all of you, the speaker, and also all the participants. So see you tomorrow, 10 minutes before 10.
Yeah. So now I can take my coffee break. It's 10:30 in Hawaii for me. So
nice time.
Okay. See everybody. That was a lot of fun today, Lionel. I like it.
Nice shirt.
Hey, really, I'll have a new one on Thursday. Tomorrow I'm wearing an LFD t-shirt. Okay. See you later.
Bye, everyone. Leonard can go have his dinner at this point.
No, still a lot of time for dinner. It's South America. It's South America.
You are not old enough to have dinner at like 4 yet.
No, if you go to a restaurant here, the last year when Dave and Enrico were here, one day I asked Dave, "What time is good for you to go to dinner?" And he said, "Well, I don't know, 6:30, 7." I called to the restaurant and I say, "No, the first time that we serve is 8:30." So,
wow.
It's the opposite of here. So.
See you tomorrow then.
Awesome.
See you tomorrow. Bye, everyone.
Bye.
Bye.
Bye. See you.
Chao, Germán. Nos vemos.
Chao. Así que va bien la cosa.
Creo que va todo bien, Germán. Sí, está saliendo, creo que... Tú paraste el...